ಮೇಡಮ್ ನಿಮ್ದಾದ್ಮೇಲೆ ಮತ್ತೊಂದು ಸೆಷನ್ ಇರೋದ್ರಿಂದ ದಯವಿಟ್ಟು ನಮಗೊಂದು 10 ಮಿನಿಟ್ಸ್ ಬಿಫೋರ್ 10 ಮಿನಿಟ್ಸ್ ಅಂದ್ರೆ 4 o'clock ಮೊದಲೇ ನಮಗೆ
ನಂದು 30 to 35 minutes ಅಂತ ನಾನು ಅನ್ಕೊಂಡಿರೋದು.
ಶೋರ್ ಮ್ಯಾಮ್.
So I think I can wind up by then.
Yes. ಶುರು ಮಾಡೋಣ ಮೇಡಮ್.
ಶುರು ಮಾಡೋಣ.
ಸೊ ಗುಡ್ ಆಫ್ಟರ್ನೂನ್ ಎವ್ರಿಬಡಿ, ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರಗಳು. ಸೊ ಇಂದಿನ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಡಾಕ್ಟರ್ ಶಾರದಾ ಮೇಡಮ್ ರವರು ನ್ಯೂರಲ್ ಕಂಟ್ರೋಲ್ ಅಂಡ್ ಕೋಆರ್ಡಿನೇಷನ್ ಎಂಬ ವಿಷಯದ ಮೇಲೆ ಉಪನ್ಯಾಸ ನೀಡಲಿದ್ದು ಅವರನ್ನು ನಮ್ಮ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಸ್ವಾಗತಿಸುತ್ತಿದ್ದೇವೆ. ವೆಲ್ಕಮ್ ಮ್ಯಾಮ್.
ಥ್ಯಾಂಕ್ ಯು ಸಿದ್ದೇಗೌಡ್ರೆ.
ಸೊ ಅದೇ ರೀತಿ ನಮ್ಮ ಅಕಾಡೆಮಿಯ ಅಧ್ಯಕ್ಷರಾದಂತಹ ಪ್ರೊಫೆಸರ್ ಅಯ್ಯಪ್ಪನ್ ಸರ್ ಅವರನ್ನು ಕೂಡ ಸ್ವಾಗತಿಸುತ್ತಾ ಮತ್ತು ಈ ಒಂದು ವಿಜ್ಞಾನ ಪ್ರತಿಭಾ ಶೋಧನೆ ಯೋಜನೆಯನ್ನು ಆಯೋಜಿಸಿ ನಮಗೆ ದಿನನಿತ್ಯವೂ ಸಲಹೆ ಸೂಚನೆಗಳನ್ನು ನೀಡುತ್ತಿರುವ ನಮ್ಮ ಅಕಾಡೆಮಿಯ ಮುಖ್ಯ ಕಾರ್ಯನಿರ್ವಾಹಕ ಅಧಿಕಾರಿಗಳಾದಂತಹ ಡಾಕ್ಟರ್ ರಮೇಶ್ ಸರ್ ಅವರನ್ನು ಕೂಡ ಸ್ವಾಗತಿಸುತ್ತಾ ಹಾಗೂ ಇಂದು ಹಾಜರಾಗಿರ್ತಕ್ಕಂಥ ನನ್ನ ಎಲ್ಲ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೂ ಹಾಗೂ ಸಹೋದ್ಯೋಗಿ ಮಿತ್ರರು ಮತ್ತು ವಿವೇಕ್ ವೃಂದದವರನ್ನು ಕೂಡ ಸ್ವಾಗತಿಸುತ್ತಾ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಒನ್ಸ್ ಅಗೇನ್ ವೆಲ್ಕಮ್ ಮ್ಯಾಮ್.
ಧನ್ಯವಾದಗಳು ಸಿದ್ದೇಗೌಡ್ರೆ. ಥ್ಯಾಂಕ್ಸ್.
ಸೊ ತಾವು ಹಲವಾರು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮಗಳನ್ನು ನಮ್ಮ ಅಕಾಡೆಮಿಗೆ ಈಗಾಗಲೇ ನೀಡಿದ್ದು, ಸೊ ತಮ್ಮ ಕಿರು ಪರಿಚಯವನ್ನು ಈಗಾಗಲೇ ಮಾಡಿಕೊಟ್ಟಿದ್ದು ಕಾಲಾವಕಾಶ ಕಡಿಮೆ ಇರೋದ್ರಿಂದ ಸೊ ಓವರ್ ಟು ಯು ಮ್ಯಾಮ್.
ಥ್ಯಾಂಕ್ ಯು ಸೋ ಮಚ್. ಅದೊಂದು ಅವಾಯ್ಡ್ ಮಾಡಿದ್ರಿ. ಓಕೆ, ಥ್ಯಾಂಕ್ ಯು. ವೆರಿ ಗುಡ್ ಈವ್ನಿಂಗ್ ಟು ಒನ್ ಅಂಡ್ ಆಲ್. ಆಲ್ಮೋಸ್ಟ್ ಈವ್ನಿಂಗ್ ಅಂದ್ಕೊಳ್ತೇನೆ. ಸೊ ಒಂದು ಇಂಟ್ರೆಸ್ಟಿಂಗ್ ಟಾಪಿಕ್ ಸಿಕ್ಕಿದ್ ನಂಗೆ ನಿಜವಾಗ್ಲೂ ಖುಷಿ ಆಯ್ತು, ನ್ಯೂರಲ್ ಕಂಟ್ರೋಲ್ ಅಂಡ್ ಕೋಆರ್ಡಿನೇಷನ್. ಸೊ ನಾವ್ ಯಾವಾಗ್ಲೂ ಇದ್ ಮಾಡಿರ್ತೇವೆ, ಬಟ್ ಟ್ರೈಂಗ್ ಟು ಟೆಲ್ ಇಟ್ ಟು ಸ್ಟೂಡೆಂಟ್ಸ್ ಮೇಕ್ಸ್ ಅಸ್ ಲರ್ನ್ ಮೋರ್ ಅಂಡ್ ಮೋರ್ ಅಬೌಟ್ ಇಟ್. ನಮಗೂ ತುಂಬಾನೇ ವಿಷಯ ತಿಳಿಸ್ಕೊಡುತ್ತೆ ಇದರಲ್ಲಿ. ಅದಕ್ಕೆ KSTಗೆ ತುಂಬಾನೇ ಥ್ಯಾಂಕ್ಸ್. ಸೊ ಲೆಟ್ ಅಸ್ ಜಸ್ಟ್ ಗೋ ಅಹೆಡ್. ನ್ಯೂರಲ್ ಕಂಟ್ರೋಲ್ ಅಂಡ್ ಕೋಆರ್ಡಿನೇಷನ್. ಸೊ what is this control and what is this coordination? Okay, slides move ಆಗ್ತಿಲ್ಲ. Okay, yeah, coordination ಅನ್ನೋದು ನಾವು ಯಾವಾಗ್ಲೂ ಕೇಳ್ತಾನೆ ಇರ್ತೀವಿ. ಸೊ ಇಲ್ಲಿ what we are referring to is organs particularly. So any two organs or more organs, when they try to interact together to end up with a particular function. So that is what we define coordination as, the process through which two or more organs interact with each other and complement the function of one another. So regular agi bodily itharaddu neditane irutte. It's not that only one organ acts and you end up with one particular activity or one particular function. So it is always a coordination of more than one organ, which usually ends up with one particular activity. Like what we are going through now, maybe what I am trying to present, or what you people are trying to listen. So all these are functions which have been synchronized, and in body you have many such functions which goes in the synchronized manner itself. Most of the activities what occurs in a human body, for example, is usually because of the coordination majorly between the nervous system and the endocrine system with the other parts of the body. Okay, so neural system and the endocrine system na major systems anta helteve. Neural system nimge gottu, it's the network of all nerves. So ಪ್ರತಿಯೊಂದು ಪಾಯಿಂಟ್ ಇಂದಾನು ಕನೆಕ್ಷನ್ಸ್, ನರ್ವ್ ಸ್ಟಿಮ್ಯುಲಸ್ ಇದ್ದೇ ಇರುತ್ತೆ. ಸೊ ನಿಮ್ಮಲ್ಲಿ ಎಲ್ಲೇ ಮುಟ್ಕೊಂಡ್ರು ಕೂಡ ನಿಮ್ಗೆ ಇಮಿಡಿಯೇಟ್ ಆ ಫೀಲ್ ಗೊತ್ತಾಗುತ್ತೆ. ದಟ್ ಇಟ್ ಸೆಲ್ಫ್ ಸೇಸ್ you have very connections of these nerves at every nook and corner of the body. Every point to point connections irutte. ಅಷ್ಟೇ ವೇಗವಾಗಿ ಆ ರೆಸ್ಪಾನ್ಸಸ್ ಒಂದರಿಂದ ಒಂದಕ್ಕೆ ಟ್ರಾನ್ಸ್ಮಿಟ್ ಆಗಿ ಯು ಎಂಡ್ ಅಪ್ ವಿತ್ ದಟ್ ಪರ್ಟಿಕ್ಯುಲರ್ ಫೀಲ್ ವಾಟ್ ಯು ಆರ್ ಫೀಲಿಂಗ್, ದಟ್ ದ ಫೀಲ್ ಆಫ್ ಟಚ್. ಹಾಗೆ ಎಂಡೋಕ್ರೈನ್ ಸಿಸ್ಟಮ್ ಏನ್ ಮಾಡುತ್ತೆ, ಇದು ಇಟ್ ರಿಲೀಸಸ್ ಸರ್ಟನ್ ಎಂಡೋಕ್ರೈನ್ ಹಾರ್ಮೋನ್ಸ್, ಕೆಮಿಕಲ್ಸ್. ಓಕೆ, ಸೊ ಈ ಹಾರ್ಮೋನ್ಸ್ ಮುಖಾಂತರ ಇದೇ ತರದ ನೆಟ್ವರ್ಕ್ ನು ಕ್ರಿಯೇಟ್ ಮಾಡುತ್ತೆ. So most of the functions in the human body are interrelated between the neural system, the endocrine system, and the other organs of the body particularly. So ಈ ಚಿತ್ರದಲ್ಲಿ ಹೇಳೋ ಹಾಗೆ ಒಂದಕ್ಕೊಂದು ತುಂಬಾನೇ ಇಂಟರ್ಲಿಂಕಿಂಗ್ ಇರುತ್ತೆ. ಕೋಆರ್ಡಿನೇಟೆಡ್ ಆಗಿ ವರ್ಕ್ ಮಾಡಿದ್ರೆ yes, it is a healthy body. If there is something happens and it is not going in a coordinated manner, it means the person is under disease. That means he is not normal ಅಂತಾನೆ ಅರ್ಥ. So coordination ಅನ್ನುವಾಗ ಮಾಮೂಲಾಗಿ ಕೊಡೋ ಎಕ್ಸಾಂಪಲ್ ಫಿಸಿಕಲ್ ಆಕ್ಟಿವಿಟಿ. So maybe we are walking or running or cycling, like how this person is doing here. Any physical activity what we do involves some kind of muscular activity. So ಈ ಆಕ್ಟಿವಿಟಿಗೆ ಎನರ್ಜಿ ರಿಕ್ವೈರ್ಮೆಂಟ್ ಇದ್ದೇ ಇರುತ್ತೆ, ಎನರ್ಜಿ ಡಿಮ್ಯಾಂಡ್ ಕ್ರಿಯೇಟ್ ಆಗುತ್ತೆ. ಸೊ ಎನರ್ಜಿ ಯುಟಿಲೈಸ್ ಮಾಡ್ಕೊಂಡೆ ಆಕ್ಟಿವಿಟಿ ಆಗೋದು. ಸೊ ಎನರ್ಜಿ ಯುಟಿಲೈಸ್ ಆಗ್ತಿದೆ ಅಂದ್ರೆ ಆಕ್ಸಿಜನ್ ಡಿಮ್ಯಾಂಡ್ ಜಾಸ್ತಿ ಆಗತ್ತೆ. ಸೊ ದಟ್ ಮೀನ್ಸ್ ನೀವೀಗ ಒಂದು ಸ್ವಿಫ್ಟ್ ಬ್ರಿಸ್ಕ್ ವಾಕ್ ಹೋಗಿ ಬಂದ್ರಿ ಅಂತ ಇಟ್ಕೊಳ್ಳಿ, ಅಥವಾ ಒಂದ್ ರನ್ ಮಾಡಿ ಬಂದ್ರಿ ಅಂತ ಇಟ್ಕೊಳ್ಳಿ. ನ್ಯಾಚುರಲಿ ಏನಾಗಿರುತ್ತೆ, ಎದುರು ಬಿಡ್ತಾ ಇರ್ತೀವಿ, ರೇಟ್ ಆಫ್ ರೆಸ್ಪಿರೇಷನ್ ಇನ್ಕ್ರೀಸಸ್, ಎದೆ ಬಡ್ತ ಜೋರಾಗತ್ತೆ, ಹಾರ್ಟ್ ಬೀಟ್ ಇಸ್ ಆಲ್ಸೋ ಇನ್ಕ್ರೀಸ್, ಆಮೇಲೆ ಎಲ್ಲ ಕೆಂಪಗಾಗ್ಬಿಟ್ಟಿರ್ತೀವಿ. ಸೊ ಅಂದ್ರೆ ಬ್ಲಡ್ ಫ್ಲೋ ಕೂಡ ಎಲ್ಲಾ ವೆಸಲ್ಸ್ ಗಳಲ್ಲೂ ಬ್ಲಡ್ ಫ್ಲೋನು ಇನ್ಕ್ರೀಸ್ ಆಗಿರುತ್ತೆ. ಸೊ ಇದು ಯಾಕಪ್ಪ ಆಗುತ್ತೆ ಅಂದ್ರೆ ಈ ಆಕ್ಟಿವಿಟಿ ಎಲ್ಲ ಕೋಆರ್ಡಿನೇಟೆಡ್ ಆಗಿ ಇವೆಲ್ಲ ನಡೀತಿರೋದಕ್ಕೆ ಯು ಆರ್ ಏಬಲ್ ಟು ಡೂ ದಟ್ ವಾಕ್ ಆರ್ ಯು ಆರ್ ಏಬಲ್ ಟು ಡೂ ದಟ್ ರನ್ ದರ್. ಸೊ ಇದು ನಿಲ್ಸಿದ ತಕ್ಷಣ ಒಂದ್ ಗ್ರಾಜುಯಲ್ ಆಗಿ ಎಲ್ಲ ನಾರ್ಮಲ್ಸಿಗೆ ಬರುತ್ತೆ. ಸೊ ಇದನ್ನೇ ನಾವು ಹೇಳೋದು ಅಂದ್ರೆ ಇವೆಲ್ಲ ಆರ್ಗನ್ಸ್ ಕ್ಲೀನ್ ಆಗಿ ವರ್ಕ್ ಆಗಿ ಕೋಆರ್ಡಿನೇಟೆಡ್ ಮ್ಯಾನರ್ ಅಲ್ಲಿ ಮಾಡಿದ್ದಕ್ಕೆ ಯು ಎಂಡ್ ಅಪ್ ವಿತ್ ದಟ್ ಪರ್ಟಿಕ್ಯುಲರ್ ರಿಸಲ್ಟ್ ಆಫ್ ದಟ್ ಫಿಸಿಕಲ್ ಆಕ್ಟಿವಿಟಿ. ಸೊ ದಿಸ್ ಇಸ್ ಜಸ್ಟ್ ಸಿಂಪಲ್ ಎಕ್ಸಾಂಪಲ್. ಈ ತರ ದೇಹದಲ್ಲಿ ನಮಗ್ ಗೊತ್ತಿಲ್ಲದೇನೆ ಎಷ್ಟೋ ಆಕ್ಟಿವಿಟೀಸ್ ನಡೀತಾ ಇರುತ್ತೆ, ಇವು ಕೂಡ ಕೋಆರ್ಡಿನೇಟೆಡ್ ಮ್ಯಾನರ್ ಅಲ್ಲೇ ಹೋಗ್ಬೇಕಾಗಿರುತ್ತೆ. ನಾವು ಟುಡೇ ವಿ ಆರ್ ಸ್ಪೀಕಿಂಗ್ ಅಬೌಟ್ ನ್ಯೂರಲ್ ಕಂಟ್ರೋಲ್ ಅಂಡ್ ಕೋಆರ್ಡಿನೇಷನ್, ನಮ್ಮ ನರ್ವಸ್ ಸಿಸ್ಟಮ್ ಬಗ್ಗೆ. ಬಿಫೋರ್ ವಿ ಗೋ ಟು ದ ನ್ಯೂರಲ್ ಆಕ್ಟಿವ್ ಕೋಆರ್ಡಿನೇಷನ್ ಅಥವಾ ಇಂಪಲ್ಸಸ್ ಹೇಗೆ ನಮ್ಗೆ ಇದೆಲ್ಲ ಫೀಲ್ ಆಗುತ್ತೆ ಅನ್ನೋದ್ರ ಬಗ್ಗೆ ವಿಲ್ ಕ್ವಿಕ್ಲಿ ಗೋ ಥ್ರೂ ದ ಬೇಸಿಕ್ಸ್ ಆಫ್ ಅವರ್ ನರ್ವಸ್ ಸಿಸ್ಟಮ್ ಆರ್ ದ ನ್ಯೂರಲ್ ಸಿಸ್ಟಮ್ ವಾಟ್ ವಿ ಕಾಲ್. ಈ ನರ್ವಸ್ ಸಿಸ್ಟಮ್ ಅಲ್ಲಿ ಅದರ ಕಣ ಅಂದ್ರೆ ಬೇಸಿಕ್ ಸ್ಟ್ರಕ್ಚರ್ ಅಂದ್ರೆ ನ್ಯೂರಾನ್ಸ್. ಸೊ ನ್ಯೂರಾನ್ಸ್ ಆರ್ ನರ್ವ್ ಸೆಲ್ಸ್, ನಿಮಗೆ ಗೊತ್ತೇ ಇದೆ. ಸೊ ಇದಕ್ಕೆ ದ ಮೇಜರ್ ಎಬಿಲಿಟಿ ಇಸ್ ಯಾವುದೇ ಸಿಗ್ನಲ್ ನ ರಿಸೀವ್ ಮಾಡ್ಕೊಳ್ಬಹುದು, ಅದನ್ನ ಹಾಗೆ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡ್ಬೋದು. ಸೊ ವಿ ಕಾಲ್ ದಮ್ ಆಸ್ ಹೈಲಿ ಸ್ಪೆಷಲೈಸ್ ಸೆಲ್ಸ್ ವಿಚ್ ಕ್ಯಾನ್ ರಿಸೀವ್ ಅಂಡ್ ಟ್ರಾನ್ಸ್ಮಿಟ್ ಡಿಫ್ರೆಂಟ್ ಕೈಂಡ್ಸ್ ಆಫ್ ಸಿಗ್ನಲ್ಸ್. ಈ ನರ್ವಸ್ ಸಿಸ್ಟಮ್ you find different types of nervous system in all kinds of living beings. ಎಲ್ಲ ಜೀವಿಗಳಲ್ಲೂ ಇದ್ದೇ ಇರುತ್ತೆ, ಬಟ್ ಇರೋ ರೀತಿ ಬೇರೆ ಇರುತ್ತೆ. ಲೋಯರ್ ಇನ್ವರ್ಟಿಬ್ರೇಟ್ಸ್ ಅಂದ್ರೆ ಹೈಡ್ರಾ ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಅಲ್ಲಿ ಏನ್ ಹೇಳ್ತೀವಿ ನಾವು, ಹೈಡ್ರಾದಲ್ಲಿ ನಿಮ್ಗೆ ಒಂದು ಸಿಂಪಲ್ ಆದ ಸ್ಟ್ರಕ್ಚರ್, ದ ಆರ್ಗ್ಯಾನಿಸಮ್ ಇಟ್ ಸೆಲ್ಫ್ ಇಸ್ ವೆರಿ ಸಿಂಪಲ್. ಸೊ ಯು ಹ್ಯಾವ್ ನೆಟ್ವರ್ಕ್ ಆಫ್ ನ್ಯೂರಾನ್ಸ್, ನರ್ವ್ ಸೆಲ್ಸ್ ಇರುತ್ತೆ, ಆಯ್ತಾ. ಸೊ ಇದ್ರದ್ದೇ ಸಣ್ಣ ನೆಟ್ವರ್ಕ್ ಇರುತ್ತೆ. ಸೊ ವಿತ್ ವಿಚ್ ಇಟ್ ಕ್ಯಾನ್ ಪರ್ಫಾರ್ಮ್ ಇಟ್ಸ್ ಆಕ್ಟಿವಿಟಿ. ಅಲ್ಲಿಂದ ಹುಳುಗಳು ಇನ್ಸೆಕ್ಟ್ಸ್ ಗೆ ಹೋದ್ರೆ ನಾವು ಇಟ್ಸ್ ಅ ಲಿಟಲ್ ಬೆಟರ್ ದೆ. ಸೊ ನಿಮ್ಗೊಂದು ಪುಟ್ಟ ಮೆದ್ಲಿರತ್ತೆ, ಅದು ಗ್ಯಾಂಗ್ಲಿಯಾಗಳಿರ್ತವೆ, ನರ್ವಸ್ ಟಿಶ್ಯೂಗಳಿರ್ತವೆ. ಸೊ ಇವೆಲ್ಲಾನು ಅದರಲ್ಲಿರುತ್ತೆ. ಅದೇ ಮೇಲಿನ ಮೋರ್ ಡೆವಲಪ್ಡ್ ಆರ್ಗ್ಯಾನಿಸಮ್ಸ್ ಲೈಕ್ ವರ್ಟಿಬ್ರೇಟ್ಸ್, ಯು ಹ್ಯಾವ್ ಅ ಮಚ್ ವೆಲ್ ಡೆವಲಪ್ಡ್ ನರ್ವಸ್ ಸಿಸ್ಟಮ್ ವಿಚ್ ಯು ಕ್ಯಾನ್ ಎನ್ಕೌಂಟರ್. So this is another picture showing the same, where you can see in the humans, you have the brain, the spinal cord, the nervous system, what we all describe. Whereas when you look into worms like the earthworm, it also has a very small brain and some segmental kind of nerves which you encounter. So these are flatworms. Even in flatworms, you will be able to see the nerve trunk, the transverse nerves and the brain. And this is a squid, mollusk ಅಂತ ಏನ್ ಹೇಳ್ತೇವೆ ಅದು. ಇದ್ರಲ್ಲೂ ಕೂಡ you have ganglions and nerves. Okay. So that means to say, if they have to perform a certain activity, they have to receive certain signals and they have to transmit certain signals. And for this, we really require the nerve network, which we refer to as our neural system. So coming to human nervous system. So in human nervous system, we classify the human nervous system into two major parts. One is the central nervous system, another is the peripheral nervous system. Central nervous system, it's like a CPU. So andre adu it is usually made up of the brain and the spinal cord, the major organs of your central nervous system. Vertebrates alli they say it is hollow and in the dorsal surface adralli gangliaಗಳು ಇರೋದಿಲ್ಲ. ಆದರೆ ಇನ್ವರ್ಟಿಬ್ರೇಟ್ಸ್ ಅಲ್ಲಿ ಇದು ವೆಂಟ್ರಲ್ ಸರ್ಫೇಸ್ ಅಲ್ಲಿ ಇರುತ್ತೆ, ಮತ್ತೆ ಇದರಲ್ಲಿ ಗ್ಯಾಂಗ್ಲಿಯಾಗಳು ಇರುತ್ತೆ. ಸೊ ಪೆರಿಫೆರಲ್ ನರ್ವಸ್ ಸಿಸ್ಟಮ್ ಅಂದ್ರೆ ಆಲ್ ದಟ್ ನರ್ವ್ಸ್ ವಿಚ್ ಕನೆಕ್ಟ್ಸ್ ಟು ಯರ್ ಸೆಂಟ್ರಲ್ ನರ್ವಸ್ ಸಿಸ್ಟಮ್. ಸೊ ದಟ್ ಮೀನ್ಸ್ ಟು ಸೇ ಫ್ರಮ್ ಎವ್ರಿ ಪಾರ್ಟ್ ಆಫ್ ದ ಬಾಡಿ, ವಾಟ್ ಎವರ್ ದ ನರ್ವ್ ಇಸ್ ಕನೆಕ್ಟಿಂಗ್ ವೆದರ್ ಟು ದ ಸ್ಪೈನಲ್ ಕಾರ್ಡ್ ಆರ್ ಟು ದ ಬ್ರೈನ್, we put them under the peripheral nervous system. So you have cranial nerves, you have the spinal nerves. And it is this which controls your voluntary functions of the body, particularly any human body for that matter. And yes, this is another picture depicting the same. As I told you, you have the central nervous system made up of the brain and the spinal cord. And as I told you, it is the site where all the information ಎಲ್ಲೇ ಪೆರಿಫೆರಲ್ ಸಿಗ್ನಲ್ ಇಂದ ಅಂದ್ರೆ ನಿಮ್ಮ ಕಾಲ ಬೆರಳ ತುದಿಯಿಂದ ಒಂದು ಸಿಗ್ನಲ್ ಹೋಯ್ತು ಅಂದ್ರೆ ನೀವೇನೇ ಟಚ್ ಮಾಡಿದ್ದು ಫೀಲ್ ಆಗ್ಬೇಕು ಅಂದ್ರು ಇನ್ಫಾರ್ಮೇಶನ್ ಪ್ರಾಸೆಸ್ ಆಗಿ ಅದ್ರ ಅನಲೈಸ್ ಆಗೋದು ಮೆದುಳಲ್ಲಿ. ಸೊ ವಿ ಕಾಲ್ ಇಟ್ ಆಸ್ ದ ಸೈಟ್ ಆಫ್ ಇನ್ಫಾರ್ಮೇಶನ್ ಪ್ರೊಸೆಸಿಂಗ್ ಆರ್ the major control of the body. So before we go much in detail with it, we'll quickly go through what forms the peripheral nervous system. Peripheral nervous system, I told you, contains all the nerves which are linked to your central nervous system. So that means to say it is a good big network of nerves which can link to your spinal cord or link to your brain. So among these nerves, we describe two types of nerves, one as afferent fibers or another as efferent fibers. Afferent andre the one which carries information to the brain. That means whatever the nerves which will sense the stimulus, okay, any stimuli for that matter, maybe it is the vision, maybe it is the auditory stimulus, or maybe it is the touch stimulus. Once it senses the stimulus, so the nerves which transfer it from the sensory neuron to the CNS, we call it as afferent fibers, that is which carries the message to the central nervous system. Efferent fibers, once the message is reached through afferent fibers, so the brain will process it and it will send message as to how you have to react to this stimulus. So we call those as motor neurons also. They take all the nerves which transmit signals from the brain to the target organ, wherever it is. So, okay, or to the motor neuron, which gives the indication as to what reaction has to be formed. We call them as efferent fibers. That means to say, imagine you are touching a hot object there. So once you touch an object there, the feel of the touch is transferred through afferent fibers to your central nervous system. And once the central nervous system processes and analyzes it through the afferent fibers, it indicates whether what the feel of hot or the feel of cold or the feel of normality, or whether you have to remove your hand or whatever action you are supposed to do there. So efferent fibers will convey the message to motor neurons. Motor neurons are the ones which induce the efferent action also. So from central nervous system to the concerned peripheral nervous system, the information is passed through your efferent fibers. So broadly, we divide the peripheral nervous system into two type divisions: somatic neural system and autonomic neural system. Somatic neural system andre they carry the impulses from the central nervous system to the skeletal muscles, whereas your autonomic neural system will transmit information from the central nervous system to involuntarily organs or involuntary movements or involuntary activities, or through autonomic neural system. Voluntary activities or through your somatic neural system. Okay. So further, this is autonomic neural system is classified into sympathetic neural system and parasympathetic neural system. So other than these, you also have one which we usually come across called as the visceral neural system. So visceral neural system is also part of your peripheral neural system. This also has nerves, fibers, ganglia and plexus which are involved. But this involves all the impulses which travel from the CNS to the visceral organs and vice versa. Okay, that which forms part of your autonomic nervous neural system also. So broadly, we can classify the human neural system into central neural system and peripheral neural system. Central neural system is made up of your brain and your spinal cord, whereas the peripheral neural system is made up of your it consists of your somatic and the autonomic neural system. The somatic neural system, I told you, majorly involves all the voluntary actions which happens through your skeletal muscles, whereas autonomic neural system is the activities or impulses which are conducted for the involuntary actions which happens in the body. So this again is classified into sympathetic and parasympathetic neural system. So to tell about sympathetic and parasympathetic neural system, this is a very nice picture. So sympathetic neural system is usually the flight or fight response what we tend to see. That means it prepares the body for any typical strenuous kind of physical activity. So, but still here, the event what we expect to occur will tend to happen here. Whereas in the parasympathetic nervous system, what actually happens is, we call it as the rest and digest symptom there. That means to say, which usually happens without much strenuous activity. So here you can see the sympathetic nervous system is usually associated with the function of all these activities which are there. Usually when we say the flight or the fight response, it is the adrenaline which is stimulated of all this, whereas the parasympathetic nervous system, which usually tends to happen without much of strenuous activity getting involved there. So let us come back to the basic structure of our neuron. To understand how the nerve functions or this kind of network acts, first you should know how a neuron acts. So neuron is what we refer to as the structural and the functional unit of a nervous system. So though when we call it as a structural and functional unit or the basic unit of your neural system, naturally you can't see it with your naked eye. So it has to be a microscopic structure. And these, as I already told you, are specialized cells which can detect, receive and transmit different kinds of stimuli. Stimuli can be anything, as I told you earlier. So stimuli may be a sudden touch response or a smell response or a visual stimuli, whatever it is. Okay, these can detect, receive and transmit the signals. So whenever we say a neuron, yes, it consists of three major parts. So one which we refer to as the cell body. This is your cell body. Okay. These are the dendrites and these are what we refer to as axons. Cell body is nothing, just like any other cell. You have the cytoplasm, you have the cell organelles there. So with the nucleus, in addition to this, you also have some granules which are present in the cell or in the cytoplasm, which we refer to as Nissel's granules. Okay. And dendrites, when we say, it is typically the branches. That means to say some short fibers at the tip of the cell body will tend to branch out continuously, giving a tree-like appearance, okay, which emerges out of the cell body. So it is just an extension of the cell body, what you are seeing here. So the contents remain the same. They also will have the granules and the other cell contents which are being present. So usually these dendrites are involved in transmitting impulses towards the cell body. That is, to take the impulse from outside towards the cell body, it is through the dendrites. So next we move on to the axons. So axons, you can see they are very long fibers. Okay. So it is these fibers. So the distal end of these fibers, we say they have a knob-like projection. Okay, a bulb-like projection, which is referred to as the synaptic knob. It is at these sites they will come in contact with another dendrite. Okay, so that it can transmit the impulses from one cell onto another cell. Okay, so here particularly, so the synaptic knob to transmit impulses involves some chemicals. So these chemicals will be seen in vesicles, and there are some closed structures which we call as vesicles. So through this information or impulses will be formed from transmitted from one nerve to the other. So we usually, the gap between one nerve to the other or to the muscle, we usually refer to as neuromuscular junction or interneural junction there. That area we call as a synapse. So nerve impulses are transmitted from the cell body through the axon to the synapse. Okay. So this is involved in transmitting the impulse from the cell body to the outside, whereas dendrites are involved in transmitting impulses from outside to inside. Okay. So now, you know, they have a cell body, they have a dendrite, and they also have axons. So based on the number of dendrites and the axons originating from a single cell body, the neurons are divided into three major categories. So one, which is called as multipolar, where you have only one axon but many dendrites there. You see this in more in cerebral cortex. Another called as bipolar, where you have only one axon, only one dendrite, not many as you see here. Okay. And unipolar, where the cell body has only one axon, you don't see any dendrites there. So this is usually unipolar structures are seen only in the embryonic stage. Okay. So you have one more picture representing the same. I'll just quickly go through that. So bipolar neurons, as telling you, so they have two processes. Unipolar, name itself suggests one process. Multipolar, when they have more. That means to say, as the first one, as I told you, multipolar neurons has one axon and two or more dendrites being formed there. Okay, so this I told you is more in the cerebral cortex. Unipolar neurons or unipolar neurons are seen with only one axon from it. So this is usually encountered in the embryonic stage, as I told you. So this is called a pseudo unipolar neuron because you tend to, this is a single neuron which is trying to go ahead. So because it gives the appearance of two neurons, two axons arising from it, we call it as pseudo unipolar here. Bipolar neurons have two processes, one axon and one dendrite there. Okay, so this is what we see in most of the body. Particularly, you tend to encounter this, as I actually explained to you earlier. So in the retina of the eye, okay, and in the olfactory epithelium, you encounter bipolar neurons very commonly. So again with axons, we describe nerve fibers as myelinated nerve fibers, non-myelinated nerve fibers. What does this mean? So myelinated andre, it has something on it. Myelin anno kotirutte. So that means to say myelinated fiber is more encountered in your spinal and the cranial nerves particularly. This axon, if you see, you have something surrounding it here, right? So these are the Schwann cells. Okay. Schwann cells, these cells in some of the nerves, they will envelop this axon and they will round up all over the axon there. Okay. So giving the appearance of one sheath being formed on it, one layer being formed on it. Okay. So this is what we refer to as myelin sheath. Okay. When a Schwann cell envelops completely around the axon. So you will have many Schwann cells going all through the length of your axon, and in between these gaps, what you see in between the myelin sheaths or between the Schwann cells is what we usually describe as nodes. That is the node of Ranvier. That is what we refer to as. In non-myelinated fiber, there also you have Schwann cell, but remember it is not enclosing your axon completely. Okay, it does not form a covering around your axon completely. It is present on your nerve cell or it is present on your axon for sure. So this is more seen in autonomic and somatic neural systems, the non-myelinated nerve fiber, whereas the myelinated nerve fiber is more seen in your spinal and the cranial nerves. So now you know about the neuron, what is a myelinated nerve, what is a non-myelinated nerve, and also the different types of neurons what we tend to encounter. So once you know the structure of the nerve, how is it? Yes, through the nerves, the impulses are transmitted or the stimulus is transmitted. Or in local terms to say, the message is transmitted. So how is this possible? So we call neurons as excitable cells because the membrane of the neurons, they say, are in a polarized state. That means to say there is the nerve membrane, what is there for your neuron, the plasma membrane, they are selectively permeable to certain ions there. Okay, so that means they have ion channels across the membrane. So we describe two kinds of conditions when we describe a neuron. One, a resting condition when there is no impulse being transmitted, or once there is a stimuli and they start transmitting impulse, that means to say they are in action, we call it as action potential. So we describe two types of state, one as the resting potential and the other as the acting potential. So resting potential, when we say, As I told you, it is not conducting any impulse. Then what happens here? So usually they say the axonal membrane is more permeable to the potassium ions, but not to your sodium ions in this state. Okay. And they're also not permeable to negatively charged proteins which are present in the axoplasm. Axoplasm refers to the cytoplasm of your axon, typically what is being present. So naturally, when it is no impulse is being transmitted, what happens is the neuronal membrane, the neuron will have high concentration of potassiums because it is more permeable to potassium ions. So you'll have more of potassium ions and less of sodium ions. Okay. So, but outside the axon, what happens is because more of potassium ions are inside, outside you have a low concentration of ions, but more of sodium ions. That means to say you have a concentration gradient which is being created. So across all the resting membranes, so whenever you have a nerve membrane there, so this keeps happening. So you have the sodium potassium pump which is being maintained. So usually when you say sodium potassium pump, if you should remember, there is for every 3 ions of sodium going out, 2 ions of potassium are taken in. Okay. So what happens here is because the outer surface of the axon will have positive charge. That means because it has more of potassium ions there. So inner surface will become negatively charged. So we call it as the nerve cell is in a polarized state. So as I told you, we describe 2 types of potential, resting and acting potential. I told you. Resting potential is the electric potential difference what you see across the resting plasma membrane. So we call it as resting potential. Whenever an impulse or stimulus is given at that particular site, there will be some activity in use. That means the polarity tends to get a little altered. So we call it as action potential. Okay. So how is it that the nerve impulses are conducted? So whenever there is a stimuli applied at a particular site, so even on the axon at a particular site, when a stimuli is there, it becomes permeable to sodium ions. If not, remember in a resting state, only potassium ions enter in, sodium ions go out. Sorry, sodium ions are not allowed in. So when stimulus is given, sodium ions also enter in. So because sodium ion enters in, the outside of the membrane will become negatively charged, inner side will become positive charge. So reversal of polarity will be there. So that means to say the membrane enters into a depolarized state there. Okay. So this difference, this electric potential difference, which is produced across the plasma membrane at the site is what we call as action potential, and how this gets transmitted from one to other is what we call as nerve impulse. So now you have at this particular site, yes, you have an impulse being generated. Okay, and then it is being transmitted. There is alteration of polarity. Now this site will become stimulus for the neighboring area, again to get depolarized. So like this, it keeps on transmitting. The resting state becomes an active state, and it moves on to become a depolarized state. And this movement of ions, or this movement of is what we refer to as movement of nerve impulses particularly. So this is also depicted in pictures, this picture. See, normally resting state first what happens, you have more of positive ions here, negative ions to the inside. So negative charge inside, positive charge on the outer surface. But once a stimuli is applied over here, so once a stimuli is applied, sodium ions tend to enter in. So once sodium tends to enter in, it becomes unstable, depolarized state, because the charge gets altered. So this keeps, once the charge gets altered here, so this acts as a stimuli to the neighboring site. So, and this continues all along the length of your axon and simultaneously between the nerves. Okay. So that is how an impulse is being transmitted. Now, how impulse can also be transmitted from one neuron through to another through junctions, what we refer to as synapses. I told you junction from one neuron to another is also referred to as synapses earlier. So when we say a synapse, that means to say you have one nerve ending here, one nerve starting here. So we call this as presynaptic area. This is a postsynaptic area if the impulse is transmitted from this direction to this direction. So a synapse means This is formed by membranes of your presynaptic neuron and the postsynaptic neuron, where you might see a gap there, okay, or you might not see any gap. They might be very closely linked also. So this gap is what we call as the synaptic cleft also, okay, and the junctions is referred to as synapses. So we describe two types of synapses, electrical synapses and chemical synapses in transmission of impulses. When we say electrical synapses, what actually happens is the membrane of this presynaptic neuron and postsynaptic neuron will be very, very close to each other. Okay. So this usually they say happens quite rarely in humans, but it's like a transmission of signals which happen from one to the other. So the potential gradient, what is being created, is transferred across the synapses to the other nerve. So it is just like how the impulse is conducted along one axon, what I told you, it will continue with the other as such. Okay. So, but they say this transfer of impulses is much, much faster than chemical synapse, which can happen. Now, let us see what is a chemical synapse. So, chemical synapse, name itself suggests there are certain chemicals which are involved in transmission of impulses. So, I told you at the tip of your axon, you have a bulb-like structure, okay, which we call as the synaptic knob. And here, this is rich in vesicles, okay. So these are called as the synaptic vesicles. So these synaptic vesicles will have certain chemicals called as neurotransmitters which are present within them. So when an impulse will arrive at the terminal, okay, it will, what happens is once the impulse reaches here from the dendrites through the cell body through all through your axon, so this synaptic vesicles will move towards the membrane and they'll fuse with the membrane there, releasing the chemicals, that is the neurotransmitters. So into the synaptic cleft, I told you here, because in electrical synapse there is very close association, you might not be able to see the synaptic cleft. So it is just the charge transfer. Whereas in chemical synapse, the gap between the neurons will be there and it will be filled with certain fluid. So we call it as the synaptic cleft there. So once they are released, this will join to some receptors which are present on the next membrane, that is the postsynaptic membrane. Postsynaptic membrane is part of your dendrite of the next nerve cell there. Okay. So once they bind there, again this binding will create alteration in the ion channels there, and again a new potential develops, and again this is transferred across this nerve cell. So this impulse might either be excitatory or an inhibitory kind of impulse. So it is better described here, maybe from one nerve to the other. So the junction I told you is called as synapse, okay, or from this is for the electrical synapse, and this is for your chemical synapse. Okay, for in chemical synapse, you have the chemicals associated, the synaptic vesicles releasing the neurotransmitters which binds to receptors and then cause changes in the ion channels or creates a concentration gradient which happens. So like this, from one nerve to the other, the chain continues. Okay, and the signals are transmitted just like how electric signals are transmitted. Okay. So coming to the central nervous system. So in the central nervous system, finally it should reach the brain, right? So the brain and spinal cord are part of your central nervous system. So we call it as the command and control, or the CPU, where the processing of the information happens. So all the voluntary movements are controlled by this. Okay, the balance, the human behavior, many activities are controlled by it. So we call it as the site for many of the activities like vision, hearing, speech, memory, anything. So this brain, is covered by the skull, okay, and you have the cranial meninges there, okay. Meninges, or you have three layers of cranium meninges, which we describe: the dura mater, the arachnoid, and the pia mater. So the outer membrane is what we call as the dura mater. The arachnoid is a very thin membrane layer. Pia mater is the one which comes in contact with your brain tissue, okay. So in the brain, we describe three parts: forebrain, midbrain, and hindbrain. So I won't go in detail with it. So the forebrain, is made up of the cerebrum, the thalamus, and the hypothalamus. Okay. The cerebrum is the major part of your brain there. So you have a cleft which originates when divides this into two halves particularly, and we call this as the right hemisphere and the left hemisphere, cerebral hemisphere. So these are connected by a tract of fibers there, which we call as the corpus callosum. The layer of cells which covers the cerebral hemisphere is what we refer to as the cerebral cortex. Okay, and the cerebral cortex is what we also call as the gray matter because it appears gray. Gray because you have more concentration of neurons in this, so it tends to get the grayish appearance. Okay. So this cerebrum has many areas, the motor areas, the sensor areas, and some areas which are not completely motor or not completely sensory areas also. And we call these areas as association areas, the larger regions. Okay. And these association areas are also associated with functions like intersensory associations or memory and communication. So white matter gets its name because wherever you have the fibers connected with the myelin sheath, so this you see more in the cerebral hemisphere, okay? So inner part of it, it gives a whitish appearance. So we call it as white matter. The outer will be grayish, so it goes as gray matter there. You have the thalamus, okay? And the hypothalamus also being present. Thalamus, the cerebrum is typically covers your thalamus, okay? So thalamus is encompassed by the cerebrum, but it is a major center for sensory and motor signaling. Hypothalamus is, just as the name suggests, below the thalamus there. Okay, you can see here, just below the thalamus is your hypothalamus. And this has major centers for controlling your body temperature or your urge for eating or drinking. And it also secretes certain hormones which you refer to as hypothalamic hormones. So your cerebral cortex has many functional divisions associated based on its function. I'll not go much in detail with it. So remember, hypothalamus is the one which maintains the body temperature and acts like a thermostat. So body temperature adjusts according to the environmental temperatures there. And this also controls the pituitary. You also have a structure called the hippocampus, which is part of our limbic system, which is majorly important for our memory and learning. When we say limbic system, so it is a group of structures which are in the inner parts of your cerebral hemisphere, deep inside, like the amygdala, the hippocampus, and everything. So usually these are the centers which control your emotions. Okay. So when we say the emotions, particular limbic system, along with your hypothalamus, they say are involved in regulating your sexual behavior also, or any expression of your emotional reactions, maybe fear or anger or whatever it is being expressed there. In your midbrain, that is the region between your thalamus, okay, thalamus or hypothalamus of the forebrain, and the pons which you encounter of your hindbrain. So hindbrain is made up of pons, cerebellum and medulla, you know that. So you have a structure called as cerebral aqueduct, which usually passes through yeah, you tend to see it here. The cerebral aqueduct, which passes through the midbrain particularly. And you also have your corpora quadrigemina, which are round swellings, which you tend to encounter particularly in the dorsal portion of your midbrain, what you encounter. And this midbrain usually associated with vision, audition, eye movement or body movement. So we also describe the main brain stem. Brainstem, though we say hindbrain is made up of pons, cerebrum, and medulla, your brainstem also involves your part of your midbrain, pons, and medulla. So it connects the brain and the spinal cord is what we refer to as brainstem. Coming to your hindbrain, as I told you, pons, your cerebrum, which is being present, and the medulla oblongata, what we refer to as. Pons is usually the fiber tracts which cover the different regions of the brain, whereas cerebellum typically has a convoluted surface. You can clearly appreciate here, okay? Because the major reason for this convolutions is providing more space so that you can accommodate more number of neurons there. So medulla is the one which is connected to your spinal cord, okay? So these also control many actions like your respirations, cardiovascular reflections, and gastric secretions also. So now you know how the nerves impulses are transmitted from the nerves. So the brain is the central area which has the major centers for this. So how these actions, or you tend to see the sensory actions which tend to happen between, or the reactions what we tend to see, the sensory reception and processing of the impulses what we encounter. We also describe certain actions or reflex actions or involuntary actions. So reflex actions are what tends to happen involuntarily when you encounter certain kinds of stimulus. That means to say you feel a stimulus and involuntarily, without your knowledge, you tend to see a action happening there. Just like you see in this picture, a person is touching a flame there, because whenever you come across this, whenever there is heat, your hand immediately is pulled back. Why is it this happens? Okay. So any action which occurs involuntarily, okay, so in response to a stimulus, but also involves here part of CNS, we call it as reflex action. So in the reflex pathway, we describe the afferent neuron and the efferent neuron. Okay. So just like your afferent and efferent fibers. So afferent neuron is one which receives the signal. Once it feels it, so immediately it transfers this red impulse being transmitted to your brain, okay, to your CNS. Once it reaches the CNS, CNS tells what? Remove your hand immediately through your afferent neurons. So immediately this is given to the muscle which supplies to the relevant muscle, and your hand is withdrawn. So this is the reflex pathway what we tend to describe. And this stimulus and response is what we call as reflex arc. Maybe the hand withdrawal reflex or the knee jerking movement suddenly when you touch over something. Okay. So all these are reflex actions. Now just a simple example of how your eye ends up with identifying colors or whatever visual signals what you tend to develop. So you have sense organs. The nose has olfactory in the sense of smell. Tongues have taste buds, which gives us the taste. Like this, you have many receptors. So here I'm just taking the example of your eye. So eye, you know the major structure of your eyeball. You have three layers: the sclera, the cornea, and the choroid, which we describe. Sclera is this a thin external layer what you are encountering, see, which is made up of connective tissue. Okay. Usually you will have a dense connective tissue here, but as it goes further, it um, uh, tends to thicken or thin out. Okay. So in the cornea is the front portion of your sclera, is what we refer to as the cornea. And the choroid is the middle layer what you encounter here. Okay. It has many blood vessels, so you tend to see it giving a little bluish tinge there. Okay. So I won't go much into the structure of the eye. I'll quickly pass through. Let me concentrate more on the impulse. Okay. So choroid layer, I told you, is thin in the posterior part, particularly two-thirds of the posterior part. But as it comes further in front, it tends to become a little thick and it forms your ciliary body there. Okay. Like what you are seeing here. Okay. The ciliary body. So the ciliary body continues to form pigmented and opaque structures called as iris as it goes ahead. The ciliary body forms your iris. Okay. And the eyeball also has a lens which is held with the support of ligaments. Okay. And attached to your ciliary body there. And in front of the lens, so the aperture, what is surrounded by the iris, is what we refer to as the pupil. So the muscle fiber of the iris will control the diameter of your pupil there. Okay. And the inner layer is what we refer to as your retina. Okay. The typical retinal layer what you are encountering. So this inner layer of your retina has your cells. Okay. The ganglion cells, the bipolar cells, and the photoreceptor cells. When we say photoreceptor cells, we describe rods and cones, two types of cells which have proteins which are sensitive to light. Okay. And these proteins are what we refer to as photopigments. So phototopic vision is what we call as daylight vision. That is what you're seeing as colored. Okay. This is because of the cones. Whereas this phototopic vision, that is whenever there is dim light, whatever you're seeing is what we call as twilight vision or phototopic vision. And this is because of the rods which are present. Okay. So these rods have a purplish red protein called as rhodopsin. Okay. This is also referred to as visual purple and is majorly a derivative of vitamin A. So in human eye, you have three types of cones which are there for color vision. Cones, you know, are for color vision. So these three types of cones also have, each one of them have unique pigments which respond to red, green and blue lights. So combination of these cones and the pigments will help you to analyze or see different colors. If all these three are equally stimulated, you usually see them as white light. So this is another picture of the retina up close there, where you're seeing the rods and the cones. Okay, typically the cone shaped and the rod shaped structures there. So it also has your ganglion cells, the bipolar cells, okay, and certain pigmented epithelium. So another cross section of your retina showing the same, the ganglion cells, the ganglion cell layer, the bipolar cell layer, okay. So all these are associated with giving you the actual vision what you are seeing. So at this site, okay, you have the optic nerve which leaves the eye, and the blood vessels also entering here at a point which is near the posterior pole of your eyeball. So we have an area here because you don't have any photoreceptor cells. We call this as the blind spot. Okay. So at this posterior pole, a little lateral to your blind spot, you have a particular area which you call as macula lutea. So this is an yellowish pigmented area, and here you see a small pit which we call as fosse. Okay, the typical fosse, which is a thin portion of retina there because you have more of cones which are densely packed. Okay, so this area is the one which they say gives us maximum visualization, or the resolution of what you are seeing is more at the this fosse. So we also describe aqueous humor and vitreous humor. Aqueous humor is the space between your cornea and the lens. Okay. The chamber is called as aqueous chamber. The fluid field is called as aqueous humor. Okay. It's a thin watery fluid. Whereas here the vitreous chamber behind the lens until the retina, we call the area as vitreous chamber or vitreous body. This is filled up with a gel-like substance which we call as vitreous humor. Okay. So this optic nerve is the one which is associated with transmitting signal from the eye to the visual cortex which is present in the brain. What happens here? So once light enters, visible wavelength of light, once it enters the eye, particularly this gets focused on the retina. Okay. So through the cornea and the lens, it enters the retina. And here, this retina is made up of rods and cones, you know that. It creates impulses or potentials. Concentration gradient potential is created here. And this impulse get transmitted. When this is created, the rhodopsin which is present here is split. Okay. So into retinol and opsin. Okay. Retinol is a vitamin A portion, opsin is the protein portion. So once this is split there, the opsin structure gets altered and the membrane permeability also gets altered here. So in the photoreceptor cells, that is in the rods and cones there, you have many potential differences which happens. Ion potentials keep changing. And this will create a signal which will generate again an action potential in the ganglion cells, okay, through the bipolar cells which are present in the wall of the retina. And these are transmitted through your optic nerves to the visual cortex in the brain. So once it reaches the brain, you have the visual cortex there. So which will analyze this impulse what is brought there. And based on either your earlier memory or based on experience, it will tell you what image it is. So you will perceive it so. Similarly with the ear, quickly we will go through this. So here, ear, has two major functions, not only hearing, also maintaining the body balance. You have the outer ear, the middle ear, and the inner ear. Outer ear majorly made of pinna, and your external auditory canal, which we call as auditory meatus, also a canal-like structure, ending up with your tympanic membrane. Okay. In addition, you have some hairs and wax secreting glands there. Okay. In the middle ear is what we refer to with your ossicles, that is the malleus, the incus, and the stapes, which are associated. And this will open to the cochlea, which is your inner ear particularly. You have a tube which we call as the eustachian tube, which will connect your middle ear to the pharynx so that the pressure is equalized on both sides. So the inner ear, particularly when we say, has labyrinth. It's called as labyrinth, and it has two parts, the bony part and the membranous part there. The fluid is referred to as endolymph and perilymph. The coiled portion is what we call as cochlea. So the space above the scala, okay, media is what we call a scala vestibuli. This is the scala vestibuli and this is your scala tympani. So your organ of Porti is usually seen in the basilar membrane there, which has some hairs and auditory receptors. These play a major role in transmitting signals. Okay, above these hairs is your tectorial membrane. Okay, above the hair cells, you see the tectorial membrane. So inner ear also has vestibular apparatus which is being present. Okay, above the cochlea, you tend to see them. And you have some semicircular canals called as saccule and the atricle. So yeah, let me not go much into this. Yeah, this is again describing your scala vestibuli, scala tympani, and the vestibular membrane being associated with it. What happens when we hear? This is what I would like to more concentrate on. So once a sound is received, okay, through from the pinna, it will be directed towards your ear canal. Okay, so once they reach this ear canal, okay, these waves which are picked up, they hit on your tympanic membrane, which you call as your eardrum, and it tends to vibrate. Okay, so once it vibrates, this vibration is passed on to your ossicles, the malleus incus and the stapes, and the same vibrations will go and hit into your oval window there. Okay, typically entering into the cochlea, so transmitting these vibrations to the cochlear fluid. So they say once it hits the oval window, here itself it is magnified many a times. So what vibrations are present in the tympanic membrane, once it reaches your oval window, it is almost 22 times more amplified than it struck your tympanic. Okay. So once it reaches your oval window there, enters the fluid of your cochlea, typically. So their wave, because it's a fluid being present there, waves are created in the lymph, whatever is there, and you tend to see ripples developing in this basilar membrane. Okay. So naturally, waves, once they're created, you see ripples, and the ripples, what happens, will go on to bend your hair cells which are being present here and press it against your tectoral membrane. So usually the vibrations, they say, are picked up by the hair cells which are present in the organ of Porti and transmitted across the neurons to your auditory nerve fibers. So what actually happens is, because of the ripples created in the fluid what is there, so you tend to see bending of the hair cells, pressing it against the tectorial membrane, creating an impulse, okay, in the afferent neurons particularly. So once the impulse is created in the afferent neurons through the auditory nerves, okay, it enters into your auditory cortex, which is present in the brain, and just similar to how your eye signals are analyzed and the sense sound vision recognized, here also the auditory cortex in the brain will analyze the impulse, and based on the previous experience or the memory, it will help you recognize the sound. So these are two major examples, the eye and the ear stimulus, what is being taken. I think that should be helpful for you to know how the neural coordination occurs so that you end up with responding to the stimuli. So with this, I would like to wind up. Let me see if there are any questions.
Dear students, ask any doubts and any questions about today's
ಚಾಟಲ್ಲಿ ಏನೋ ಹಾಕದಂಗಿತ್ತು, ಯಾರೋ ಚಾಟಲ್ಲಿ ಏನೋ ಹಾಕದಂಗಿತ್ತು.
ಇಲ್ಲ ಇಲ್ಲ ಮೇಡಮ್, ಅದು ನಾನೇ ಇದು ಮಾಡಿದೀನಿ.
ಹೌದಾ? ಓಕೆ ಓಕೆ ಓಕೆ.
ಯಾವುದು ಕ್ವಶನ್ಸ್ ಇಲ್ಲ. ಸೊ ಏನೋ ಕಳಿಸಿದ್ರೆ ನಾನು ಲೇಟರ್ ಆನ್ ಐ ಸೆಂಡ್ ಥ್ರೂ ಮೇಲ್ ಮ್ಯಾಮ್.
That should be fine. That should be fine. I think I'll stop sharing then.
ಮತ್ತೊಂದು ಕಾರ್ಯಕ್ರಮ ನೀವು ಮುಗಿಸ್ತಾ ಇದ್ದಂಗೆ ಶುರು ಆಗುತ್ತೆ ಮೇಡಮ್.
ಗೊತ್ತಾಯ್ತು, ಅದಕ್ಕೆ I would like try entering it.
Anyway, thank you madam.
Okay, hope it was helpful for the students. Thank you so much. Thanks Siddhev. Thanks to KSTA also. Thank you very much.
ಥ್ಯಾಂಕ್ ಯು. ತಾವು ಕೊಟ್ಟಂತ ಇಂದಿನ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಮಕ್ಕಳಿಗೆ ಹಲವಾರು ಮಾಹಿತಿಗಳನ್ನ ತಿಳಿಸ್ಕೊಟ್ಟಿದ್ದೀರಾ. ಸೊ ನನ್ನ ವೈಯಕ್ತಿಕವಾಗಿ ಹಾಗೂ ನಮ್ಮ KST ವತಿಯಿಂದ ಧನ್ಯವಾದಗಳು ಮೇಡಮ್.
ಧನ್ಯವಾದಗಳು ತಮಗೂ ಕೂಡ. ಥ್ಯಾಂಕ್ ಯು.
ಥ್ಯಾಂಕ್ ಯು ಮೇಡಮ್. ಅವ್ರು ಯಾರು ಮಕ್ಕಳು ಯಾರು ಕ್ವಶನ್ಸ್ ಕಳ್ಸಂದ್ರೆ ಐ ಸೆಂಟ್ ಥ್ರೂ.
ನನಗೆ ಮೇಲ್ ಅಲ್ಲಿ ಕಳಿಸಿ ಐ ರೆಸ್ಪಾಂಡ್. ಓಕೆ
ಪ್ಲೀಸ್ ಒಂದು ರಿಕ್ವೆಸ್ಟ್ ಮೇಡಮ್, ಈಗ ನೀವೇನು ಟಾಪಿಕ್ ನೀವು ಟೀಚ್ ಮಾಡಿದಿರೋ, ಸೊ ಆ ಟಾಪಿಕ್ ಮೇಲೆ ನನಗೊಂದು 3 to 4 pages ನೋಟ್ಸ್ ನಾನು ದಯವಿಟ್ಟು.
ನಿಮ್ಗೆ ಓಕೆ ಇನ್ಫಾರ್ಮೇಶನ್ ಕಳಿಸ್ಬೇಕಾ? ಓಕೆ ಹೆಲ್ತ್ ಸೆಂಟರ್.
ಅದು ನಾವು ಒಂದು ಕಿರುವತ್ತಿಗೆ ಟೈಪ್ ಅಲ್ಲಿ ಒಂದು ಬುಕ್ ಮಾಡಿಸಿ ಮಕ್ಕಳುಗಳಿಗೆ ಹಂಚ್ತಾ ಇದೀವಿ.
ಓಕೆ ಓಕೆ ಓಕೆ
ದಯವಿಟ್ಟು ಮರಿಬೇಡಿ, ಕಳಿಸ್ಕೊಡಿ.
ಕಳಿಸ್ಕೊಡ್ತೀನಿ ಸಿದ್ದೇಗೌಡ್ರಿ. ಥ್ಯಾಂಕ್ ಯು.
ಒನ್ ಸೆಕೆಂಡ್. ಥ್ಯಾಂಕ್ ಯು ಮ್ಯಾಮ್.
ಓಕೆ ಐಲ್ ಲೀವ್.
ಸೊ ಈಗ PPT ಶೇರ್ ಮಾಡಿದ್ದೀನಿ ಸರ್. ಈಗ ಸರ್ ಅಲ್ವಾ? ಸೊ ನೀವು ಡೈರೆಕ್ಟ್ ಈಗ ಒಂದು ನಿಮಿಷ ಕರೆದು ಬಿಡ್ತಿದ್ದಾರೆ ಸರ್, ಕರೆಕ್ಟ್ ಆಸ್ಪೆಕ್ಟ್.
ಸರ್ ಇಲ್ಲ ಗೊತ್ತಿಲ್ಲ.
ಸರಿ, ಇಲ್ಲ ಶೇರ್ ಮಾಡಿದ್ದೀನಿ.
ಏನಾಗಲ್ಲ ಕ್ಯಾಮರಾ ಎಲ್ಲಿ ಸರ್ ವೆಬ್ಸೈಟ್ ಕಾಣ್ತಾ ಇಲ್ಲ. ವೆಬ್ಸೈಟ್ ಕಾಣ್ತಾ ಇಲ್ಲ. ವೆಬ್ಸೈಟ್ ಅಷ್ಟು
ಕರೆಕ್ಟ್ ಇರುತ್ತೆ ಮಾಮೂಲಿ ಬಂದಿರ್ಬೋದು.
ಕರೆಕ್ಟ್ ಆಗಿದೆಯಾ?
ಸರ್ ಶೇರ್ ಕರೆಕ್ಟ್
ಕರೆಕ್ಟ್ ಆಗಿ ಶೇರ್ ಮಾಡ್ತಾರೆ. ಇದು ಹೋಗ್ತಾ ಹೋಗ್ತದೆ.
ತಡಿ ಒಂದು ನಿಮಿಷ ಸರ್, ಹೇಳ್ಕೊಡೋ ಒಂದು ನಿಮಿಷ.
ಸರ್ ಇದು ಈ ಬಟನ್ ಒತ್ತಿದ್ರೆ ಆಯ್ತು. ನೆಕ್ಸ್ಟ್
ಅಲ್ಲ, ಕ್ಯಾಮ್ರಾ ಬರ್ತಾ ಇದೆ.
ಕ್ಯಾಮ್ರಾ ಆನ್ ಮಾಡಿ.
ಒಂದು 2 ನಿಮಿಷ ನಾನೇ ಮಾಡ್ತೀನಿ.
ಇಲ್ಲ ನಿಮ್ಗೆ ಕಾಣಬಹುದು.
ಇಂಟ್ರೊಡಕ್ಷನ್ ಕೊಡ್ಬೇಕಲ್ಲ ಸರ್? ರೇಟ್ ಕೊಡೋದಾ? ಚೇಂಜ್ ಅಲ್ವಾ? ಸೊ ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರಗಳು. ಇಂದಿನ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಡಾಕ್ಟರ್ MS ಜೋಗ ಸರ್ ಅವರು ಉಪನ್ಯಾಸ ನೀಡಲಿದ್ದು, ಅವರನ್ನು ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಸ್ವಾಗತಿಸುತ್ತಿದ್ದೇನೆ. ಹಾಗೂ ನಮ್ಮ ಅಕಾಡೆಮಿ ಅಧ್ಯಕ್ಷರಾದಂತಹ ಪ್ರೊಫೆಸರ್ ಅಯ್ಯಪ್ಪನ್ ಸರ್ ಅವರು ಕೂಡ ಸ್ವಾಗತಿಸುತ್ತಾ, ಅದೇ ರೀತಿ ನಮ್ಮ ಮುಖ್ಯ ಕಾರ್ಯನಿರ್ವಹಣಾಧಿಕಾರಿಗಳಾದಂತಹ ಡಾಕ್ಟರ್ ರಮೇಶ್ ಸರ್ ಅವರನ್ನು ಕೂಡ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಸ್ವಾಗತಿಸುತ್ತಿದ್ದೇನೆ. ಸೊ ಡಾಕ್ಟರ್ MS ಜೋಗದ್ರವರು ತಮ್ಮ ಬೋಧನಾ ವೃತ್ತಿ ಜೊತೆಗೆ ಸಂಶೋಧನಾ ವೃತ್ತಿ ಎಲ್ಲರ ಜೊತೆಗೆ ಶಿಕ್ಷಕರಾಗಿ ಸುದೀರ್ಘ ಬೋಧನಾ ಅನುಭವವನ್ನು ಹೊಂದಿದ್ದು ಮತ್ತು ಉತ್ತಮ ಶಿಕ್ಷಕ ಪ್ರಶಸ್ತಿಯನ್ನು ಪಡೆದಿರುತ್ತಾರೆ. ಪದವಿ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ಭೌತಶಾಸ್ತ್ರದಲ್ಲಿ ಕಡಿಮೆ ವೆಚ್ಚದ ಕಿರುಹೊತ್ತಿಗೆ ಹೊರತಂದು ಮೇ 9-12-2004 ಅವಧಿಯಲ್ಲಿ ಚೀನಾ ಶೆಂಜಾನ್ನಲ್ಲಿ ನಡೆದ ಹೈ ಪರ್ಫಾರ್ಮೆನ್ಸ್ ಸೆರಾಮಿಕ್ಸ್ ನ 3ನೇ ಚೀನಾ ಅಂತಾರಾಷ್ಟ್ರೀಯ ಸಮ್ಮೇಳನದಲ್ಲಿ ಭಾಗವಹಿಸಿ ಅದರಲ್ಲಿ ಅಧ್ಯಕ್ಷರಾಗಿ ಕಾರ್ಯನಿರ್ವಹಿಸಿದ್ದಾರೆ. ಹಾಗೂ ಡಾಕ್ಟರ್ ಜೋಗಲ್ ಸರ್ ಅವರಿಗೆ ಫೆಬ್ರವರಿ 28, 2003 ಕರ್ನಾಟಕ ಸರ್ಕಾರದಿಂದ ಸರ್ ಸಿ.ವಿ. ರಾಮನ್ ಯುವ ವಿಜ್ಞಾನ ಪ್ರಶಸ್ತಿಯನ್ನು ಕೂಡ ನೀಡಲಾಯಿತು. ಅವರು ಐಎಪಿಟಿಯ ಪ್ರಧಾನ ಕಾರ್ಯದರ್ಶಿ ಹಾಗೂ ಕಾರ್ಯಕಾರಿ ಮಂಡಳಿಯ ಅಧ್ಯಕ್ಷರಾಗಿ ಕಾರ್ಯನಿರ್ವಹಿಸಿದ್ದಾರೆ. ವಿಜಯಪುರ ಅಕ್ಕಮಹಾದೇವಿ ಕಾಲೇಜಿನ ವಿಶ್ವವಿದ್ಯಾಲಯದಲ್ಲಿ ಭೌತಶಾಸ್ತ್ರ ಸಂಯೋಜಕರಾಗಿ ಕಾರ್ಯನಿರ್ವಹಿಸುತ್ತಿದ್ದು ಡಾಕ್ಟರ್ ಎಂ.ಎಸ್. ಜೋಗಲ್ ರವರು ಶರಣಬಸವೇಶ್ವರ ವಿಜ್ಞಾನ ಮಹಾವಿದ್ಯಾಲಯದಲ್ಲಿ ಭೌತಶಾಸ್ತ್ರ ಪ್ರಾಧ್ಯಾಪಕರಾಗಿ, ಮುಖ್ಯಸ್ಥರಾಗಿ, ಪ್ರಾಂಶುಪಾಲರಾಗಿ ಸುದೀರ್ಘ ಸಾರ್ಥಕ ಸೇವೆ ಸಲ್ಲಿಸಿದ್ದಾರೆ. ಅಧ್ಯಯನ, ಅಧ್ಯಾಪನ ಮತ್ತು ಸಂಶೋಧನಾ ಕ್ರಿಯೆಗಳಲ್ಲಿ ತೊಡಗಿಸಿಕೊಂಡಿದ್ದಾರೆ. 2001ರಿಂದ 2002ರವರೆಗೆ ಫುಲ್ಬ್ರೈಟ್ ಶಾಲರಾಗಿ ಅಮೆರಿಕದ 3 ವಿಶ್ವವಿದ್ಯಾನಿಲಯಗಳಲ್ಲಿ ಪ್ರಾಧ್ಯಾಪಕರಾಗಿ ಹಾಗೂ ಸಂಶೋಧಕರಾಗಿ ಕಾರ್ಯನಿರ್ವಹಿಸಿದ್ದಾರೆ. ಕೆಲವು ಗಾಜಿನ ಸಿರಾಮಿಕ್ಸ್ ತಯಾರಿಕೆಗೆ ಮತ್ತು ಅಧ್ಯಯನಗಳು ಎಂಬ ಶೀರ್ಷಿಕೆಯ ಪ್ರಮುಖ ಲೇಬಲ್ಸ್ ಯೋಜನೆಯನ್ನು ಬಾಬಾ ಅಣುಶಕ್ತಿ ಕೇಂದ್ರದಿಂದ ಅವರಿಗೆ ನೀಡಲಾಯಿತು. ಅದರಲ್ಲಿ ಅವರು ಪ್ರಮುಖ ಸಂಶೋಧಕರಾಗಿ ಹಾಗೂ ಡಾಕ್ಟರ್ ಜೆ.ಪಿ. ಕೋಟಿಹಾಳ ಬಾಬಾ ಅಣುಶಕ್ತಿ ಕೇಂದ್ರದ ಸಂಯೋಜಕರಾಗಿ ಹಲವಾರು ಹೊಸ ಸಂಶೋಧನೆ ಕೈಗೊಂಡು ಈ ಯೋಜನೆಯಲ್ಲಿ ಕೈಗೊಳ್ಳಲಾದ ವಸ್ತು ವಿಶೇಷ ಗಾಜಿನ ಸಿರಾಮಿಕ್ಸ್ ಮೆಟಲ್ ಸೀಲ್ಗಳಲ್ಲಿ ಬಳಸಲು ಹೆಚ್ಚಿನ ತಾಂತ್ರಿಕ ಪ್ರಾಮುಖ್ಯತೆಯನ್ನು ಹೊಂದಿದೆ. ಇದಲ್ಲದೆ ಹಲವಾರು ವಿಷಯಗಳ ಮೇಲೆ ಸಂಶೋಧನೆ ಕೈಗೊಂಡು ಬಾಬಾ ಪರಮಾಣು ಸಂಶೋಧನಾ ಕೇಂದ್ರದಲ್ಲಿನ ಧ್ರುವ ರಿಯಾಕ್ಸ್ ಸೌಲಭ್ಯದಲ್ಲಿ ನ್ಯೂಟ್ರಾನ್ ಸ್ಕ್ಯಾಟರಿಂಗ್ ಅಧ್ಯಯನ ಕೂಡ ಮಾಡಿ ವಿಜ್ಞಾನ ಕ್ಷೇತ್ರಕ್ಕೆ ಒಂದು ಹೊಸ ಕೊಡುಗೆಯನ್ನು ಸಹ ಕೊಟ್ಟಿರುತ್ತಾರೆ. ಓಕೆ, ಥ್ಯಾಂಕ್ ಯು ಸರ್. ಓವರ್ ಟು ಯು.
ನಮಸ್ಕಾರ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ. ತಮಗೆ ಗೊತ್ತಿದ್ದ ಹಾಗೆ ಅಷ್ಟೇ ತಮಗೆ ಗೊತ್ತಿದ್ದ ಹಾಗೆ ಇವತ್ತಿನ ದಿವಸ ದ ಲೈನ್ ಸ್ಪೆಕ್ಟ್ರಾ ಆಫ್ ದಿ ಹೈಡ್ರೋಜನ್ ಆಟಮ್ ಬಗ್ಗೆ ಮಾತಾಡ್ತಾ ಇದ್ದೇನೆ. ಇದೊಂದು ಕರ್ನಾಟಕ KST ಅವರದ್ದು ಒಂದು ಒಳ್ಳೆ ಕಾರ್ಯಕ್ರಮ. ಇದರದ ಸದುಪಯೋಗ ಬರ್ತಾ ಇಲ್ಲ. ಇದರ ಸದುಪಯೋಗ ಎಲ್ಲಾ ವಿದ್ಯಾರ್ಥಿಗಳು ಪಡೆದುಕೊಳ್ಳಬೇಕು ಅಂತ ಒಂದು ಇಚ್ಛೆಯಿಂದ ಅವರು ಮಾಡಿದ್ದಾರೆ. ಏನೇ ಆಗಲಿ, ಎಷ್ಟೇ ವಿದ್ಯಾರ್ಥಿಗಳು ಇವತ್ತಿನ ದಿವಸ ಜಾಯಿನ್ ಆಗಿದಾರ, ಅದರ ಬಗ್ಗೆ ನಾನು ಕುಲಂಕುಷವಾಗಿ ಹೇಳೋದಕ್ಕಿಂತ ಮುಂಚೆ ಕೊಟ್ಟ ಅವಧಿಯಲ್ಲಿ ನನಗೆ ಕೊಟ್ಟ ಒಂದು ವಿಷಯದ ಮೇಲೆ ಇವತ್ತಿನ ದಿವಸ ತಮ್ಮೊಂದಿಗೆ ಚರ್ಚೆಗೆ ಪ್ರಾರಂಭ ಮಾಡ್ತೇನೆ. ಬಹುತೇಕ ವಿದ್ಯಾರ್ಥಿಗಳ ಗಮನಕ್ಕೆ ತರಬಯಸ್ತೀನಿ. ಇಂಗ್ಲಿಷ್ ಮತ್ತು ಕನ್ನಡ ಎರಡೂ ಮಿಕ್ಸ್ ಮಾಡಿ ಹೇಳಕ್ ಪ್ರಯತ್ನ ಮಾಡ್ತೇನೆ. ವಿದ್ಯಾರ್ಥಿಗಳು ಗೊತ್ತಿರಬೇಕು, ಇದು ಲೈನ್ ಸ್ಪೆಕ್ಟ್ರಾ ಆಫ್ ಹೈಡ್ರೋಜನ್ ಒಂದೇಳಿ ನಾವು ತಿಳ್ಕೋಬೇಕಾದ್ರೆ ಇದಕ್ಕಿಂತ ಮುಂಚೆ ಯು ಶುಡ್ ನೋ ಅಬೌಟ್ what are the fundamental particles and how this spectra can be explained, hydrogen spectra. You know, during the early 20th century, I'm talking this during the earlier 20th century, several research activities has taken place. Unrelated developments in physics converged to provide the basis for a surprising new model of atomic structure. To understand the line spectra of the hydrogen atom, first we can understand the atomic structure. Atomic structure. Now you know, I already mentioned about the 19th century. One contribution was the collective observation from the study of optical spectra. I am using the word optical spectra. Another was Planck's explanation of the distribution of the energy of light emitted from an incandescent object. Two concepts I am mentioning here. One concept is contribution, collective observation of many things. And another is contribution of the Planck's explanation. Planck's proposal that the energy of the light depended on its frequency and was in turn used by Einstein to explain an observation known as the photoelectric effect. You know, in 19th century, many experiments they did, and experimental results they are unable to explain because the theory was not developed. So again started the many researchers they have proposed these diverse ideas along with Rutherford's discovery of atomic nucleus. You have already studied both in physics as well as in chemistry in 1911. But in 1913, Niels Bohr conceived a radically new atomic model. You see, the Bohr's role is very important to understand both classical physics as well as the quantum contribution, quantum physics. You see here the many experimental facts, including the Planck's proposal, Einstein's idea, everything is used for the explanation. Both theories successfully explained many of the puzzles. See, Bohr's theory successfully explained many of the puzzles that had arisen in the studies of optical spectroscopy. I am using the optical spectroscopy because this spectroscopy is used to understand the atomic structure. That was the beginning of a new understanding of the atomic structure, was based on observational evidence. I already mentioned in 19th century, many experiments they performed, but results were analyzed, but theory was not supporting. Spectra of light emitted are observed by the various chemical elements. You know already, if you heat a iron, you will get a greenish light. If you heat other element, different colors will get it because compositions are different. These observations were important in the development of new theories because the theoretical model had to be consistent with the observations. Initially, spectroscopy was the study of optical spectra because today we have to study the line spectra of the hydrogen atom, which results from dispersing light in component colors. You know already, whenever you put a light pass through the prism, the light disperses into different colors that we will study in the book that we jar violet, indigo, blue, green, orange, red, like that, spreading the light according to its wavelength. This idea was extended to other regions of electromagnetic spectrum. You know, electromagnetic spectrum, central visible region is there, and this side ultraviolet, this side infrared, and beyond ultraviolet X-rays, beyond X-rays, gamma rays. This already it is there in your textbooks. You have studied even in high school. If you come to this side, infrared, radio waves, microwaves, so many things we have already studied. Today's spectroscopy includes the study of spectra from different views, but we are not interested in today's class to study detail about the spectroscopy. We are interested to study this line spectra of hydrogen atom. I already mentioned 19th century work was very important, more contribution from experimental, but only theory is not cope up in those days. Later it was developed from different scientists from Rutherford, Thomson, Chadwick, and many later Planck, Maxwell's, and Einstein, these are the things. Now, why the line spectra of hydrogen is important? Because hydrogen contains only one electron, central nucleus, that already model of the atom you have studied. It is very simple to do experiment as well as to develop the theory. Why is so important? Because today we think of spectra as the fingerprints of matter. I am repeating this word, spectra as the fingerprints of the matter. One minute. The spectra as the fingerprints of the matter. Every atom or molecule emits its own characteristic spectrum of light. You see, gold is a different spectra, silver shows the different spectra, platinum shows the different spectra. Therefore, spectra as the fingerprints of the matter. Every atom or molecule emits its own characteristic spectrum of light. No two spectra are alike. They are different. We may produce spectra by heating a gas at low pressure in an electronic discharge tube. That is what we have to do today. We have to take hydrogen in a tube, to pass a electric current through that, discharge takes place, and pass that light through the prism. That is, we'll get the spectra. We have to study that. Now, before going to that part, why I'm showing this? Because in those days, they have studied the spectra. Emission spectra appear as a series of bright lines. That means in those days, scientists like Fraunhofer, he studied the spectrum of the light which is coming from the sun. Those are called the Fraunhofer lines. Similarly, take a hydrogen, heat it, take the spectra, that is called the atomic hydrogen spectra. Take a material like barium, heat it, will get the barium spectra. Similarly, mercury, heat it, will get mercury spectra. Similarly, easily you can do it for sodium, heat it, will get the sodium spectra. Now, these spectra I will show you later. Again, here in the spectra, emission spectra appears as a series of bright lines, each representing a particular wavelength. I will show you later. And similarly, another type of spectra, absorption spectra, appears as a series of dark lines. You see, whenever by heating some absorption of energy takes place, that spectra gives the line of dark lines. That's what we are getting in a light from the sun, that is called the Fraunhofer line. But in the case of hydrogen, barium, mercury, and all these things, they are the emission spectra. Now, here I already mentioned many scientists starting from Thomson. That is what he mentioned here in this slide, because Thomson discovered the electron and charge is negative. This whole thing, what is the mass is very less, and all these things I shown here. Similarly, another fundamental particle is proton. It was discovered by Goldstein. Be careful while writing the answers. Similarly, charge of proton, they have put it as positive. That was mentioned by the Rutherford. Similarly, it is heavier than the electron by 1,837. Similarly, neutron, that is discovered by Chadwick, and no charge here, and mass also you have shown here, spin is also shown here, and all these some properties we have shown. Now here I will go how these things started. This is after 19th century. The thing is why these spectral lines are important. That is, we have shown experimental evidence of spectral lines. Similarly, in physics, you study black body radiation and photoelectric effect also. These experimental evidence provides a clues to the deeper understanding of the nature and behavior of the atoms. But these clues were not readily interpreted in those days because there was no perfect atomic model. There was not understanding clearly what is there inside. But attempt was done by the Rutherford in 1911 by doing the alpha scattering experiment. Anyhow, they will mention that is not in your syllabus, alpha scattering experiment. Later, what he has done after performing those experiments, he proposed that atom had a tiny positively charged nucleus, whole mass is concentrated inside, and other volume is very free, and remaining thing is electrons orbiting, something that he mentioned. The Rutherford description of a positively charged nucleus surrounded by electrons conflicted with the classical electromagnetic theory. In those days, in classical physics, what they developed before 19th century, that theory was not with this what he proposed. If the electrons were assumed to be at rest, no possible stable configuration could be found. The electrons would just fall in toward the nucleus. If you say that electron is sitting there somewhere distance from the nucleus, after some time that will reach to the nucleus, fall to the nucleus. On the other hand, if electron is revolved in a circular orbit about the nucleus, they would undergo constant acceleration toward the center. But Maxwell's equation predict that accelerating charges would radiate away the energy, and if it loses the energy and spiral things takes place, ultimately it will spiral into the nucleus. Now, whatever he proposed, this theory giving this idea. Now, what is it? Then that keeps the atom from collapsing. But that means what halfly is correct, but the remaining thing is not correct. Therefore, many scientists, including the in 1911, or he started answering this question. What is the question? Is what he assumed that center is the nucleus, electron in a different orbit, and if you take rest, there is one problem. If you consider if it is a motion, again there is another problem, collapsing the nucleus. But Bohr was not only scientist, he was professor at the university. He is a very smart. He spent 1 year with the Rutherford and 1 year with the J.J. Thomson. You see, he worked 1 year with J.J. Thomson. You know, J.J. Thomson was so famous in those days. He was discovered the electron and all these things. And he spent some time with the Rutherford also. Later, he returned to the hometown, that Copenhagen. He combined the ideas of Rutherford. What is the Rutherford concept of nucleus? And also the concept of Planck, quantum hypothesis. And also the idea of Einstein. And he combined this all, Thomson idea, Rutherford's idea, Planck's idea, and Einstein. Later, he answered the question. Whatever the problem is there in those days about the experimental evidence and the answer by theory were not possible. Therefore, for some year, he answered by using this all theory of Rutherford, Thomson, Einstein, and Planck, by using those ideas, Bohr evaded the classical difficulties by observing that the existence of stable atoms implies that laws of classical adverbs were not appropriate for the description of the atoms and therefore need of the alteration of this Rutherford model. Therefore, Bohr began by considering the simplest case, that of positively charged nucleus with a single electron traveling in a closed orbit around it. Therefore, they considered only hydrogen atom. This model corresponds to the hydrogen atom. He further assumed that mass of the electron was negligibly small compared with the mass of the nucleus. Then he postulated the two important things. What is that important thing you study in the your syllabus? Electron revolves about the nucleus in a stationary orbit or stable. If it is a stationary, also you study that that has to obey the angular momentum condition. You know what is the angular momentum? MVR. That what is that quantum condition? Integer multiple of h by 2 pi. That is nh2 by 2 pi. Now this what is this is the first postulates of the Bohr's. Second postulates is even in the first postulates it is against the classical physics, not consistent with the classical physics. Why? Bohr assumed that while the electrons are in stationary orbit, they may be treated with the classical dynamics. Classical dynamics means including the Newton's laws, Coulomb's law, and all these things, ordinary conservation rules. But they are not consistent. Whatever it is a Bohr's hypothesis. The second is atom emits light only when the electron makes a sudden change from one energy state to another energy state. That means usually in the book they will write higher energy state is written by E2, lower energy state is written by E1. If it jumps from higher energy to lower energy means difference of energy is E2 minus E1 that emits the photon that is having the energy equal to h. That is what we study. This is the second postulate. Now, again, in the second postulates also, Bohr assumed that when radiation thus takes place, the relations between the energy radiated and the frequency is given by Planck's equation. Planck's equation is what? E equal to h nu. Now, you see here, these two postulates are very important. Those postulates are called the Bohr's postulates. Now, you should not equate the Bohr radii, Rn, with atomic radii. Bohr radii is different. Now, already given one question how to determine the Bohr radii. They will share later. You can answer that. I think some 3 problems I have given. They will share later, but you have to answer. I think some prize is there. The idea of electron orbits as circles with sharply defined radii contradicts. That means the idea is circular orbits, stationary orbits, so many things he put. They are not. It contradicts the experimental evidence. However, for hydrogen, because we are taking the single electron atom, that is hydrogen, that is good agreement exists between the predictions of Bohr's theory and experimental observations of spectra and energy levels. That is what we have to study the spectra today. Now, difficulty here, according to Rutherford, atom is spherical and all these things. It has a small positive charges. These are the Rutherford's proposition. Nucleus is surrounded by electron. Electron revolve around the nucleus with very high speed in a circular path called orbits. Now, how this energy is maintained? 2 forces are there. One is electrical. Whole atom is an electrical neutral. Why? Because electrons are negatively charged. Nucleus is positively charged. Therefore, it is a neutral atom. Electrons nucleus held together. Electrons also held together with the nucleus because some force is there between the electron and charge of the nucleus. That force is called the electrostatic force of attraction. If you take Z is the charge of the nucleus, E is the charge of the electron, distance between that is R square, and you will get the Z square by R square some equation. And also you can put some equality for 1 divided 4 pi epsilon and all these things you study that is mentioned in the book. The Rutherford model has resembles like a solar system. planetary model. You can also call it planet model. Different planets move around the sun. And therefore, sometimes Rutherford model we call it as the planet model of the atom. And here one more problem is there is an empty space around the nucleus called extranuclear part. In this part, electrons are present as the nucleus of the atom is responsible for the mass of the atom, and the extranuclear part is responsible for its volume. Now, these are the good part of the Rutherford's model, but again, so many drawbacks are there. What is the drawback is according to the electromagnetic theory of Maxwell. But in physics and 18th century and 19th century, they are dominated by Maxwell's equations, Maxwell's electromagnetic theory and all these things. And this theory is not consistent with the Rutherford. When a charged particle moves under the influence of attractive force, that is what I mentioned, Z square divided by R square, it loses its energy continuously in the form of electromagnetic radiation. Therefore, an electron in an orbit will emit radiation. As a result of this, electrons should lose energy at every turn and move closer and closer to the nucleus. This is what I mentioned earlier, spiral path or something. This is the drawback of the Rutherford model. Therefore, Rutherford's model cannot explain the ultimately, you can put it in a single word because you will write IIT and CET. You can say that Rutherford's model cannot explain the stability of the atom. If you come to the another drawback, the electrons lose energy continuously. The spectrum is expected to be continuous. You see here, electrons loses energy continuously as mentioned in the Rutherford. The expected spectrum is what? Continuous spectrum. But if you do the experiment, instead of getting this continuous spectrum, we will get well-defined lines of definite frequency. I will show that. At the end of that, I have the figure. I will show you here. At the end, I have this figure. I will show this. This is the figure I will show. Therefore, you will appreciate. I put it the last because of what is the time available, I don't know. Therefore, I put these figures are last. And here, 1 minute. These all derivations, you can do it. I use here. Are you there? Can you see here? I already mentioned about the Purana lines. That is the spectrum. Another is MCN spectrum, like atomic hydrogen spectrum. These are the lines. The line shape is like whatever the slit will take in the experiment that is there, barium, mercury, and all these things. And Pranapar is a dark lines. Anyhow, these are the figures. One minute. Now, what I am talking about the Rutherford's drawback, that instead of getting the continuous spectrum, we will get the, as per the theory, what proposed by Rutherford, it should be continuous, but that is not there. But here, if you come here, Rutherford, another drawback, you can say go here. 1 minute. This another drawback is instead of getting the continuous spectrum, we'll get the line spectrum. But you see here, now question is, I will come directly to the topic now, atomic spectrum. Spectrum means what? Whatever that impression is there on the screen, what we get, particular wavelength are analyzed through the either passing through the prism or grating, and you can call it as a spectrum. Here I show continuous spectrum. That is, here slit is there, pass through the prism. This is what we'll get it. Now again, spectrum has a two types, emission spectrum and absorption spectrum. And emission spectrum, what we'll get, when the radiation emitted from the source, for example, by heating the gas, by heating the element, or from the sun, whatever that will get, pass through the prism or to the grating or to on photographic plate, whatever the spectrum will get, that is called the emission spectrum. Example, there are again in the emission spectrum two types, continuous spectrum and discontinuous spectrum. That is the line spectrum, because what we have to understand the line spectrum. And when light from any source such as sun, bulb or any hot glowing body is analyzed by passing through prism, it is observed that it splits up into 7 different colors. That is because these colors are continuous. Merging is there at the border. Then each of them merges into the next. Hence, the spectrum is called the continuous spectrum. And again, this continuous spectrum when gas or vapors of chemical substance are heated in an electric arc or a Bunsen flame, light is emitted, I already mentioned, by hitting the iron, it get a greenish color. If the ray of this light is passed through the prism, we will get a line spectrum. The discontinuous spectrum consists of distinct and well-defined lines with the dark areas in between is called line spectrum. That is what earlier I showed. Fraunhofer lines, hydrogen atom lines, barium and all these things. They are the line spectrum. Now, here, again, if you come to here, difference between the line and band spectrum. Again, here, band spectrum and the molecular spectrum, whatever that we call, what exactly difference between line and band spectrum? The line spectrum has sharp, distinct, well-defined lines. That is what I show. Band spectrum has many closed lines are there. That is what the 7 colors. The line spectrum is the characteristic of atoms and is also called atomic spectrum. You see, Mostley's law, whatever that is there, you will study in chemistry as well as in physics. And here, you are in Bangalore or other places, they use the satellite, the special satellites are there. They will take photograph where the ores are there, gold ore, copper ore, and bauxite or zinc, something, and we'll get it from the spectrum. Those spectrums are the line spectrum. That means line spectrum is the characteristic of the atom. Gold spectrum is different than silver spectrum. Therefore, line spectrum is the characteristic of atom and is also called the atomic spectrum. And line spectrum is due to the transition of electron and atom. I will show here next after the Bohr's theory, Bohr's model. I will show how the electron jumps from higher orbit to the lower orbit. Different transition will get, different wavelength will get. And Balmer series, first Lehman series, Balmer series, and all these series we can study later after this. And band spectrum has many closed lines. Band spectrum is a characteristic of a molecules. There in the line spectrum, characteristic of an atom. This molecules contains different and NaCl, sodium and chlorine, a molecular spectrum will get. I like that. Band spectrum is a molecular spectrum. Line spectrum is an atomic spectrum. And there, line spectrum will get it due to the transition of electron from one orbit to the another orbit. Already electrons in a different orbit. They are shown in the Bohr's theory. First orbit, second orbit, and third orbit. And each orbit is given the name K, L, M, N, like that. And also there we will study the quantum mechanical number. Those are called the principal quantum number. Later we can study that. Band spectrum is given by hot metals and molecular moments. Now again, I already mentioned there is the emission spectrum and another is the absorption spectrum. And white light from any source is first passed through the solution or vapors of the chemical substance, then analyzed by the spectroscope. It is observed that same dark lines are obtained. Further, it is observed that dark lines are at the same place where color lines are obtained in the emission spectra. Now, these are the difference between the emission spectra and absorption spectra. I already mentioned what exactly difference between emission spectra and absorption spectra, and also we have discussed about the difference between line spectra and band spectra. Now, emission spectrum is obtained when the radiation from the source are directly analyzed in the spectroscope. Similar like that, but I will go to the another transition. This is what in the line spectrum we get. Whenever if you pass a hydrogen gas, pass light, you know that whenever you get the energy from higher orbit and to lower orbit, like that you can say K, L, N, N. Higher orbit energy you can give it by the lower orbit E1, higher orbit E2. The spectrum obtained, is we already know, if E2 electron jumps energy, let us take you assign energy to the higher orbit is E2, lower orbit is E1. If by chance energy, those are excited state electrons, if you come to the lower state, there is a difference of energy, that is the Bohr's second postulate E2 minus E1. And let us take from second orbit to the first orbit, the transition will get a different series, those are called Lemon series. And from another one is the Balmer series, another is the Poisson series, and Bracket series, Poppand series, Humphrey series, like that. Here in the figure I show Lehman series will get whenever electron jumps from higher orbit to the first orbit. Balmer series will get from higher orbit to the second orbit or second energy level. And Poisson series will get from higher orbit to the third level of energy. Now you see that energy in what region, again that depends on the strength of the energy, whether it comes in visible region or it comes in the ultraviolet region, whether it comes in the infrared region or near infrared region, far infrared region. That also I showed here. These transitions are wave number of all the lines will get it by the Rydberg. I already showed first attempt done by this Balmer. Balmer, he gave the equation, empirical equation, no theoretical background. But he done for the many things. He tried for hydrogen atom, he was successful. Again applied to the higher orbit, but he collected many data. Later, Rydberg collected all the data. He also put it in a very simple way, that is wave number. Wave number means reciprocal of the wavelength 1 by lambda, that equal to R, Z square is the atomic number, 1 divided by N1 square minus 1 by N2 square. Those are called the whole number. Later you study in the quantum mechanics, those are called the principal quantum number. I have shown the line diagram also, and important is Balmer series, Balmer equation and Rydberg equations. Now here, this how postulates, how it is calculated, important thing is in Bohr's, whatever that stationary orbit is there, important thing they are given is quantization, angular momentum quantization. That means MVR equal to NH by 2 pi. That is, this whole thing will study there. And important in the second postulate is A2 minus E1 equal to H nu. Already I mentioned, Bohr was so smart, he used the concept of Rutherford, Planck, Einstein, everybody's. He put it in a very simple way because he was not only a scientist, he was a professor, he was a teacher. Therefore, it was possible to put whole thing is in a pictorial manner and with some equation. Even today, the quantum mechanics is developed a lot, but even then we are using Bohr's theory, because Bohr's theory that gives the visualization for the energy level diagram and all these things. And you can calculate the radius of the orbit, that is called the first Bohr orbit. That is what you will get by equating the equation, that is the two forces, electrostatic force as well as the angular momentum. You do that, you will get the radius of the equation n square by 4 pi square. This you can derive the equation, and whatever that lowest this is the Bohr's radius. That value is 0.0529 nanometer. Nano is what? 10 raised to minus 9 meter. Now, whatever it may be, this also I given some problem. Similarly, you can calculate energy of the spectrum. To calculate the energy of the spectrum, we have to use the kinetic energy, and that is the half mu square. Similarly, energy equation, I equate to the electrostatic force and all these things. First, you write the total kinetic the energy, total energy equal to kinetic energy plus potential energy, and evaluate the energy equation. That energy equation becomes minus 2 pi square mz square e raised to square divided by n square h square. This equation, substitute the corresponding value, you will get the energy. Now, I already mentioned what is the electromagnetic and what are the limitations of the electromagnetic, and this electromagnetic theory unable to explain these things. Black body radiation, photoelectric effect, line spectra, whatever the hydrogen line spectra, that was unable to explain. Only thing is that the Balmer he gave an empirical equation modified by the Reaper by collecting the data. Those equations we are using for calculating the energy and all these things. Now here, and you can take different equations. What is the number of series? What is the spectral division? What is the wave number? What energy is there? How the equation is there? These things you can do that. How to do this derivation part? Center is the nucleus, and whole mass is concentrated there. The electron is moving with definite velocity and orbiting there, and use the electrostatic force equation, use the energy equation, calculate radius, calculate velocity, calculate energy equation, and wavelength can be calculated by using the Rydberg equation, and R is called the Rydberg constant, and that is the some special number is there, 1.0962 into 10 raised to 9, some equation is there that you can do it. But here, why person is important is Balmer. But Balmer was so smart, and Balmer used the idea of prana plasma, and that was great interest in spectroscopy. Later, attempt made by Balmer, he found a simple mathematical formula that related the wavelengths of the prominent lines in the visible and infrared spectrum, especially for hydrogen gas. Because hydrogen exhibits one of the simplest atomic spectra, that is what we have studied today, line spectra. But Balmer formula, I already mentioned, Balmer he gave an empirical equation, the wavelength lambda of hydrogen that equal to 364.56 nanometer n square divided by n square plus 2 square, where n is an integer that takes the value of n equal to 3, 4, 5, 6. That whole derivation I given here. Later they will share it. And also all textbook use the derivation and all these things. The corresponding lines observed in the visible spectrum of hydrogen are called the Balmer series. All Balmer series are kept in the visible region. And using the Balmer, this equation, Balmer calculated the wavelengths of the 9 lines, 4 visible and 5 ultraviolet, that were then known to exist in the spectrum of hydrogen. I already shown that hydrogen spectrum. Balmer's formula was strictly empirical, no theory. Just he put like this. Kepler's equation is also empirical technique, and Balmer's equation is also an empirical equation, no theoretical background. That is, it was not derived from any physical model or theory of physical behavior. Instead, Balmer offered his formula simply as a mathematical relationship that was consistent with the observation. Whatever he observed experimental, he put it in some adjustment equation. There was no apparent reason why it should work, and they are not understood why he is getting the same answer what we are getting from the experiment. Even he was also not knowing. It provided amazingly precise computation of wavelength hydrogen spectrum, even less than error of 0.1% for all the 9 lines. In announcing his formula, Balmer suggested it might possibly be a special case of some here line I already showed. No, these lines you can see here. Balmer series, Lehman series, Paschen series in this figure color, and y-axis is an energy, x-axis is a wavelength. This is the figure I already showed. And from the ideas, from the data of Balmer, the another scientist, Robert Rydberg, sought to find such a formula. He tried to put a mathematical equation theoretically. He was successful, and now those equations are called the Rydberg formula. I already shown 1 by lambda means what? Wave number mu. Mu equal to 1 by lambda, that equal to r infinite 1 divided by n1 square minus 1 by n2 square. N1 is less than n2. N1 means orbit number. R0 is called the Rydberg constant. The value is and 10973731 553 per meter. Vapor constant is what? 1.097 1, 2, 3, 4, 5, 6, 7, 10 raised to minus 7. And this is what is called a vapor constant. Now, you can use this equation, you can calculate what is the wavelength of the line in Farmer series with the longest wavelength. This problem already given, you will share, you can calculate. That answer will come out to the 656 nanometer. That line is 656 nanometer means red color. 400 nanometer means blue color. And that is what you should understand. Therefore, I will get these equations by using that. Now here, just to discuss some figure. What is that? What is that? Only 8 minutes. Right, that is what I. Now this is what I discussed the nucleus. Center is nucleus, how electron is rotating, how they are equating that the charge of the nucleus and this electrostatic equation as well as those equations are already used there. And another figure how transition is there from higher orbit to the lower orbit. That is what we have done. Now here I show continuous spectrum of the sun. That is the Fraunhofer line. Very clear is there. And second is atomic hydrogen. That is what they are taken figure in those days. And these are the experimental results. In those days, these results are not explained because there was no perfect theory. Later, with the development of atomic models, starting from Dalton, starting from so many scientists, we have done that. Now here, why I am talking uh, this line spectra is a very important concept. Therefore, really it is not only important in physics, it is also important in this what you call chemistry also. Now, you should understand first this all model and all these things and how things will carefully will get an idea. Now, I already mentioned. How many times? Yes. Now here hydrogen spectrum, these all things are there. Later I already mentioned there are many theories they have developed, and by using those theories, later they explain the whole thing. You can understand just highlight how the development takes place. John Dalton, the atomic theory with spherical, because any scientific development is there not by the single person. In today's theory itself, you saw that it is starting from J.J. Thomson, starting from Rutherford, with Einstein, with everybody's idea. He used the board, he put the equation. Yes, yes.
ಆಮೇಲೆ ಲಾಸ್ಟ್ ಅವರ್ ಮಾತಾಡ್ಬೋದು.
Yes. But how many years is there?
ಮಾಡಿ ಇನ್ನೂ ಅರ್ಧ ಗಂಟೆ ತೊಂದರೆ ಮಾಡಿ ಸರ್, ನಾನು ನಿಜ ಹೇಳ್ತೀನಿ. ಆಮೇಲೆ ಚಾಟ್ ಬಾಕ್ಸ್ ಮತ್ತೆ ವಾಟ್ಸ್ಆಪ್ ಗ್ರೂಪ್ ಇವೆಲ್ಲ ಕಳ್ಸಿಕೊಡ್ತಾರೆ ನಮ್ಮ.
Yes, yes, yes. Now I think so far shown some equation and all these things, and I already put 3 problems, and they will share to the chat box. You can solve those problems. Just I will view hierarchy started this discovery in atomic structure. This whole thing is a discovery in atomic structure. John Dalton, he proposed an atomic theory with spherical solid atoms based upon measurable properties of mass. First, he has given. Next, Packer built one of the first gas discharge tubes, cathode rays tube, whatever that we call it today. And the next development by the Maxwell proposed the electric and magnetic fields, and that helped for to understand the atomic structure. Next come the Crookes tubes, because you study in the chemistry or in physics, Crookes tube, whenever you pass some discharge takes place, some spectral lines will get, and discovered cathode rays, because you heard the cathode rays. What is there at two end? You have the plate pass a gas through that, discharge takes place and all these things, that is the cathode rays, had the following uh, properties. That is the fluorescence, negative charge to object, strike, and all these things you are studied in first two. Then the important person comes the Goldstein. He used the cathode ray to study the canal rays, which had electrical and magnetic properties opposite of electron. Electron charge is given negative, and canal rays have the opposite charge, that is the positive charge. Next comes the discovery of X-ray. Now, discovery X-ray by the Rayington. These all things are helped to understand the atomic structure. Using cathode ray tube, he observed that nearby chemicals glow. Further experiment found very penetrating rays. Now, many things they have done. Here, you see here, so far we have the idea of what are the spectra, what are the types of spectra, what is emission spectra, what is continuous spectra, what is line spectra. Here is the line spectra we get it. You see, by understanding the structure of the atom, ultimately we are able to understand why the line spectra will get. How this energy is there. The given wavelength means energy you get. Wavelength is what? This is the frequency you can calculate. Later you can calculate energy. This is the sketch of Balmer lines in spectrum of atomic hydrogen. Now here, next Rington, he discovered the X-rays. That times I helped him understand the atomic structure. And Becquerel, while studying the effect of X-rays on photographic film, he discovers some chemical spontaneous decomposition and give up very penetrant rays. So many things here they have studied. Next comes J.J. Thomson. He used the cathode ray tube experimentally to determine the charge to mass ratio, E by M. Next, Rutherford. He studied the radiation emitted from uranium and thorium, and later he has shown the alpha rays. Alpha scattering experiment is very important to understand the structure of the atom. Later he built the atomic model. Next, Planck used the idea of quanta, pocket of energy, that E equal to h. Next comes the Millikan. He determined the charge of an electron. You know already, charge of an electron equal to 1.602 into 10 raised to minus 19 coulomb. And also you know the mass of 10 raised to minus 31 kg of an electron. If you want its easiest, 9.11 into 10 raised to minus 28 gram. No, with this all development, I already mentioned, science is not developed by single person. There are a lot of people they have developed. That is what in Canada it is a DASO. Without expecting anything to him, the scientists they will do for the welfare of the society. Similarly, I have given hierarchy how started the discovery, how they help for the structure of the atom, and all these things. Next comes the Rutherford. Using alpha particles as atomic bullets, probed the atoms in a piece of gold foil. He established that the nucleus was very dense. The remaining thing is only electrons cloud is there. Very small and positively charged particle. He also assumed that the electrons were located outside the nucleus. After the Rutherford, I already mentioned the line spectra is very important. Here I show the Balmer series. This line spectra is also called the atomic spectra. Line spectra is also called atomic spectra. Why atomic spectra? Here we have taken one single atom of hydrogen, electron, you take helium. If it jumps from higher orbit to the lower orbit, there is a transition is there, there is a difference of energy is there. That is the photon that we have given in the form of here line we have given. Now similarly, you can study these things separately. That is how you can put it like a CET questions or put it like a IIT questions. I already shared. So far what we have done to understand the line spectrum of hydrogen, what is the background is required? Background is required is the atomic model, atomic structure, starting from John Dalton and also composition of atom that contains electron was discovered by J.J. Thomson. Actually, J.J. Thomson discovered the electron. Later, cathode rays was discovered by William Crookes and J.J. Thomson. Discharged to so many things we have studied. Properties of X-rays, X-ray discovered, charge of electron, so many things we have studied. Proton was discovered Goldstein, and the properties of anode, and all so many things developed before from that. Another important thing is in the line spectra, why it is important. By using the line spectra, we will get a different atoms, different elements. If you want to understand where gold is available, if you want to understand where copper is available, just you have to take the spectrum of the ore. You can trace the air that is there. Even uranium is very important, and we can get it there, and by using the line spectra. Another important thing is atomic number or nuclear charge. The number of protons present in the nucleus of the atom is called atomic number. And you plot a graph, root of frequency versus the atomic number, you will get the some line. From that line, you can trace where that is there. That Moseley's law is important because that helps to trace the different ores and all these things. Like that, many things are there you can understand, and some problems I already shared. The hydrogen spectrum, what actually spectrum is there? Spectrum is the impression of produced on a photographic film when the radiation of particular wavelength analyzed through prism or diffraction grating. That is the spectrum means. And this is the atomic spectrum. Types of spectrum already are studied: emission spectrum, continuous spectrum, line spectrum. In the emission spectrum, we have the continuous spectrum and line spectrum. Absorption spectrum is an example of Fraunhofer line that is in the sun. This continuous spectrum and line spectrum, we get it from the metals and all these things. Absorption spectrum produced by the absorbed radiation is called absorption spectrum. Hydrogen spectrum is an example of line emission spectrum. Hydrogen spectrum, what today we have studied line spectrum is what? Hydrogen spectrum is an example of line emission spectrum or atomic emission spectrum. When an electric discharge is passed through a hydrogen gas at low pressure, a bluish light is emitted. That you can do it very simple experiment. This light already our officers, PhD scientists, they are displayed in the first slide how that light passes through the prism, how that line spectrum will get. That is the line spectrum of hydrogen spectrum, and this is what they have shown. And this hydrogen is an example of hydrogen spectrum is an example of line emission spectrum or atomic emission spectrum. How to calculate the wavelength or wave number? We have to use the Rydberg equation that is first started from the Palmer. And you know, at energy level diagram also I shown that is the Lehman series, and this is what I shown here. How that Palmer series will get, how the Lehman series will get, how the Paschen series will get, how Propan series will get. That is by using the this equation. And sometimes to evaluate the wavelength of various hydrogen lines, and some scientists, that scientist name is Ritz, introduced the following expression. That is mu number equal to 1 by lambda, mu by c equal to r into 1 by r one square. This r is a constant. Sometimes in loop they will write r infinite or r only they will write. This is the universal constant. This is called the Rydberg constant. Sometimes that formula is also called the Rydberg equation. But truly those equation expression is given by the Ritz, scientist Ritz. Now earlier than that, you have studied in the model. You don't think that only Rutherford is given the atomic model. Earlier than that, many people attempted, even Thomson's also, those are called plum pudding model of Thomson. And in that, a positively charged sphere and here and there electron uniformly distributed like in a watermelon. Watermelon cut it at the center, house heats are there, those are the electrons. And the remaining space you can put it as parallel. That is the plum pudding model of Thomson. That is the beginning. And earlier than that also Rutherford. Later modified by the Rutherford nuclear model by the alpha scattering. And this all today we have studied that. And next comes the so many drawback for Rutherfords. And ultimately, I already mentioned, Moore was so intelligent, so wise. He spent 1 year with Rutherford, 1 year with J.J. Thomson. He, inspired by those people, he used those concepts and put it in a very developed, modified model because he thought that without altering those models, we cannot explain experimental evidences. Therefore, use the concept of Rutherford, concept of Lens, concept of Einstein. He put a model by proposing some postulates. Those are called Bohr's postulates. Now, what are the drawback? How it is successful? Ultimately, advantage of Bohr's theory is what? Bohr's theory satisfactorily explains the spectra of species having one electron, because only one electron is the hydrogen. And helium, lithium later he extended, and he calculated the radius of the, now those are called the Bohr's radius, and that is some value you get if that problem is given. You can calculate the Bohr's radius, Bohr's velocity, and whatever that energy equations and all these things you can calculate by using the Ritz equation, and those are called the Rydberg formula and all things. Now, spectral evidence for quantization. Here, Bohr's theory starts means it is the beginning of the quantum mechanics. I am putting the beginning of the quantum mechanics because he used the concept of quantization, because he used that spectral evidence for quantization, explanation for hydrogen spectrum on the basis of Bohr's atomic model. That is, light absorbed or emitted as a result of electron changing orbits produces a characteristic absorption and emission spectrum, which can be recorded on the photographic plate as a series of lines. The optical spectrum of hydrogen consists of several series of lines called Neyman series, Balmer series, Paschen series, Brackett series, Popon series, and Humphrey series. These spectral lines were named by the name of scientists who discovered them. Now, here to evaluate the wavelength of various hydrogen lines, and this shows that equation, and we have already studied that. I'm repeating this because to well aware of this failure of Bohr model. There is so much he took all the evidence and also he gave the this Balmer series line spectrum and everything. He explained more or less most of the things were accepted, but few are failure of Bohr model is there. Bohr theory was very successful in predicting and accounting the energy line spectra of hydrogen. Bohr's theory was successful in explaining what are the line spectra we get, whether it is jumping from higher orbit to the first orbit Lehman series, jumping from higher orbit to second series Balmer series, higher orbit to the third series, that is Paschen series. He was successful in calculating those wavelengths. But problem is in hydrogen only one electron is there. If you extend to the more electrons, multi-electron system, it would explain, it could not explain the line spectra of atoms containing more than one electron. This is the failure. For hydrogen, perfectly theory and experimental was satisfied. For multi-electron like helium, lithium, if you take the transition, if you do this, it is not successful. Second is, this theory could not explain the presence of multiple spectral lines. This theory could not explain the splitting of spectral lines in magnetic field. You study, if you go to the higher studies in BSc or engineering, if you same these lines, spectral lines, you put it in a magnetic field, the splitting takes place. There are two splitting. One is a normal Zeeman effect and another is anomalous Zeeman effect. In the case of normal Zeeman effect, three splitting is there. One becomes original, another is higher, and another is lower. Similar and like this, that splitting of spectral lines not explained by the Morse model. Intensity of these spectral lines was also not explained. These are some failure is there. Whatever it may be, more than 90% successful of this whole model to explain the experimental evidence. This theory could not explain the uncertainty principle also. If you go to this higher quantum theory and all this, and this theory was unable to explain dual nature of matter as explained as it is a deeper wave nature and particle nature. Electron behave like wave as well as particle, that also not explained by Bohr's theory. No conclusion was given for the concept of quantization of energy, but he used the quantization concept in angular momentum quantization and all these things. With this, how this Bohr's line spectrum, especially here, we'll study the Balmer series and all these things. These transition is their lines in the spectrum of atomic hydrogen, and Bohr's theory is explain this for hydrogen atom clearly by using the Bohr's concept, by using the different theory, by using the Rydberg equation, by using the Balmer's equation, everything. And I given the all background of atomic model, how it is started, what are the equations are required to develop this Balmer series and all these things. This we have given. And students, those who are interested to understand the in detail, you can solve the problem. I given three problems, you can solve it. And also I given the CET type questions from different CET as well as IIT. I think scientists from PhD they will share you. If you have any question, you can write to me also, write to the KST officers, they will forward to me. I will answer you. And if you want, you can take my email jogad1952@rediffmail.com. Once again, I will repeat. My email is jogad1952@rediffmail.com. Thank you very much for patience hearing. I am very thankful to the KST officials, especially to the CEO of KST, Dr. Ramesh sir, and as well as my friend Professor Ayappan, and also other officials. Thank you one and all. Please write to me or write to the KST. I will answer. Thank you. Best of luck for it. Thank you one and all. And also I thank you to Siddharth for arranging this, all the things.
Thank you, sir. Thank you, Maharaj. Thank you very much. So today you delivered a very good, wonderful talk on line spectrum. ಸೊ ಈ ಒಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮವು ಮಕ್ಕಳಿಗೆ ಹಲವಾರು ರೀತಿಯ ಮಾಹಿತಿಗಳನ್ನ ತಿಳಿಸ್ಕೊಟ್ಟಿದೆ ಹಾಗೂ ತಾವು ಕೊಟ್ಟಂತಹ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮವು ಹಲವಾರು ಉದಾಹರಣೆಗಳ ಮೂಲಕ ಲೈನ್ ಸ್ಪೆಕ್ಟ್ರಮ್ ಅಂದ್ರೆ ಏನು, ಅದು ಯಾವ ರೀತಿ ಒಂದು ಇದರಲ್ಲಿ ಮೂಡುತ್ತೆ ಅಂತನ್ನೋದನ್ನ ಭಾರಿ ಅನ್ವೇಷಣಾ ಪೂರ್ತಿಯಾಗಿ ಮಕ್ಕಳಿಗೆ ತಿಳಿಸ್ಕೊಟ್ಟಿದ್ದಾರೆ ಸರ್. ಸೊ ನನ್ನ ವೈಯಕ್ತಿಕವಾಗಿ ಹಾಗೂ ನಮ್ಮ ಅಕಾಡೆಮಿ ಪರವಾಗಿ ಧನ್ಯವಾದಗಳು ಸರ್. ಅದೇ ರೀತಿ ನಮ್ಮ ಅಕಾಡೆಮಿ ಅಧ್ಯಕ್ಷರಾದಂಥ ಪ್ರೊಫೆಸರ್ ಅಯ್ಯಪ್ಪನ್ ಸರ್ಗೂ ಕೂಡ ಧನ್ಯವಾದಗಳು ಸರ್. ಸೊ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮವನ್ನು ಆಯೋಜಿಸಿದ ಬಗ್ಗೆ ದಿನನಿತ್ಯ ಸಲಹೆ ಸೂಚನೆಗಳನ್ನು ನೀಡುತ್ತಾ ಬಂದಿರುವ ನಮ್ಮ ಅಕಾಡೆಮಿಯ ಮುಖ್ಯ ಕಾರ್ಯನಿರ್ವಾಹಕಾಧಿಕಾರಿಗಳಾದಂಥ ಡಾಕ್ಟರ್ ರಮೇಶ್ ಸರ್ಗೂ ಕೂಡ ಧನ್ಯವಾದಗಳು ಸರ್. ಹಾಗೂ ಇಂದು ಹಾಜರಾಗಿದ್ದ ಎಲ್ಲ ವಿದ್ಯಾರ್ಥಿ ಮಿತ್ರರು ಹಾಗೂ ಸಹೋದ್ಯೋಗಿಗಳು ಹಾಗೂ ವಿವೇಕ್ ವೃಂದದವರಿಗೂ ಕೂಡ ಧನ್ಯವಾದಗಳನ್ನು ತಿಳಿಸುತ್ತಾ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮವನ್ನು ಮುಗಿಸುತ್ತಿದ್ದೇವೆ. ಹಾಗೂ ಸರ್ ಅವರು ಕೆಲವು ಸ್ಲೈಡ್ಸ್ ಮತ್ತೆ ನೋಟ್ಸ್ ಗಳು ಕೊಟ್ಟಿದ್ದಾರೆ. ನಾನು ನಿಮ್ಗೆ ವಿವೆಕ್ಸ್ ಗ್ರೂಪ್ ಮತ್ತೆ ಚಾಟ್ ಬಾಕ್ಸ್ ಎರಡಲ್ಲೂ ಹಾಕಿರ್ತೀನಿ. ತಾವು ದಯವಿಟ್ಟು ಎಲ್ಲಾ ವಿದ್ಯಾರ್ಥಿಗಳು, ಹಾಜರಾಗಿದ್ದ ಎಲ್ಲಾ ವಿದ್ಯಾರ್ಥಿಗಳು ಕೂಡ ಉತ್ತಮ ಉತ್ತರಗಳನ್ನು ಬರೆದು, ಜೊತೆಗೆ ತಾವು ಈ ಒಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮದ ಬಗ್ಗೆ ತಮ್ಮ ಅನುಭವಗಳನ್ನ ನಾನು ಕಳಿಸಿರ್ತಕ್ಕಂಥ ಗೂಗಲ್ ಫಾರ್ಮ್ ಅಲ್ಲಿ ದಯವಿಟ್ಟು ಮರೆಯದೆ ಬರೆದು ಕಳಿಸಿ ಅಂತ ಮತ್ತೊಮ್ಮೆ ನಮ್ಮ ಅಕಾಡೆಮಿ ವತಿಯಿಂದ ತಿಳಿಸುತ್ತಾ ಈ ಕಾರ್ಯಕ್ರಮವನ್ನು ಮುಗಿಸುತ್ತಿದ್ದೇವೆ. ಥ್ಯಾಂಕ್ ಯು ಸರ್. ಥ್ಯಾಂಕ್ ಯು ಅರುಣ. ನಿಮ್ದ ಸರ್ ಜಾಸ್ತಿ ಹೊಡಿರೋದು ಎಷ್ಟು ರೆಕಾರ್ಡಿಂಗು?